Good afternoon, everyone. Welcome to the day 2 of the 2022 Research Symposium, presented to you by the UP Office of the Vice President for the Academic Affairs. We are about to start our program in a few, so please text or call your colleagues, classmates, or friends to log in now in our Zoom or tune in to our live streaming via YouTube. We are also posting the links on the chat box. For those who joined us yesterday, thank you for staying and joining us this day too. If you just joined today, don't worry, as we still have 6 more presentations this afternoon. Once again, welcome to day 2 of the 2022 Research Symposium. To give us a recap of yesterday's presentations and to set the mood for today's sessions, please help me welcome the co-chair of the EIDR Monitoring and Evaluation Committee, Professor Emeritus Elsie Jimenez.
Good afternoon, everyone. Before we start, let me remind you about the poster exhibits that are available on the OVPAA website, and the link to the website is available in the chat box. For the EIDR Cycle 8, there are 13 programs or projects. A program consists of 2 or more projects. Yesterday, we witnessed the presentation of the 7 programs or projects, and today we shall listen to the presentation of the remaining 6. For session 1, on day 1, we listened to the presentation of reports in the field of material science. In session 2, we listened to topics in the areas of the humanities and the social sciences. Now, for the first project, it is entitled Zinc Air Battery, Oxygen Electrocatalyst Research and Prototype Design and Development, with project leader Dr. Ocon, in collaboration with 4 project staff members, 2 PhD students, and 11 MS students. The project is about zinc air batteries with potentially good performance, resource availability, safety, cost effectiveness, and environmental impact compared to lithium-ion batteries. Their accomplishments include several articles published in Web of Science Index journals, paper presentations in international conferences, and 2 generations of emergency metal air battery prototypes. For the second project, we listened to the polydopamine nanocoatings and nanoparticles research for target beneficial applications, with program leader Dr. Nuesca, collaborating with project leaders Dr. Yago and Dr. Yazal, 3 project staff members, 3 MS students, and 2 BS students. The projects on materials chemistry investigate the beneficial properties of polydopamine films and other biocompatible materials. It was revealed that polydopamine film deposition of polymers and other substrates had great potential for biomedical applications. The research contributes to the understanding of the synthesis of polydopamine modified micellar structure for possible targeted delivery and controlled release application. The third project is on highly luminescent and doped carbon quantum dots from fishery waste materials for potential application in photocatalytic degradation of selected dyes. This was presented by project leader Dr. Ponce, who collaborates with 3 project staff members, 1 junior faculty, 1 researcher, 2 MS students, and 3 BS students. The project aims to provide efficient bioremediation Marine resources can serve as biorefinery of chemicals and materials for the synthesis of quantum dots. The project, the project accomplishments include article on carbon quantum dots, utility model, collaboration with the University of Saskatchewan in Canada, and application for trademark termed AVAMOS for a valorization of agri-fisheries materials using opportune science. Hopefully, these works on material science will generate patents and more utility models aside from publications in Web of Science and Scopus Index journals. The fourth program is entitled Digital Development, Printing on Demand, and distribution of Sora, the graphic novel of the life of Melchora Aquino, a perception study of our nation's history, culture, struggle for independence, and identity of Filipino women in the open distance learning framework. The program leader is Dr. Alfonso, in collaboration with project leaders Dr. Bandelaria, Dr. Lumanta, Dr. Saludades, Dr. Flor, Dr. Garcia, together with 35 SORA team members. The program depicts the role of Tandang Sora and many unrecognized Filipino women in the struggle for national independence through the creation of a graphic novel, Sora, ang babaeng para sa bayan, that is aimed to popularize history. The projects include digital graphic novel as performative research, comparative study of effectiveness of the novel in printed and e-formats, effectiveness of massive upper online course or MOOC in teaching history, and social construction of individuals and collective identities in a graphic novel-based MOOC. This project can become a prototype for use in other MOOCs. The fifth project is in cinema for education, rationalizing the Philippine audiovisual industry to promote inclusive education through film literacy. The program leader is Professor De Ocampo, in collaboration with project leaders Dr. Buenviaje, Dr. Aragones, Dr. Datuwin, and Professor Delene, together with 10 project staff members and several students from various Philippine universities. So far, 4 films were produced. These are Puppet Passion, Martial Law and Philippine Cinema, Terrazilia, and Kaamado and Comrade Carlos. 5 films are in production. These are Philippine Alternative Cinema, Mangroves, Astro Pinoy, Hydrozilia, Golden Age of Philippine Cinema. The project aims to provide education instead of mere entertainment derived from cinema. Outputs and deliverables in combination with CHED project are 9 educational films, 9 ancillary materials with evaluation, and peer-reviewed publications of 3 essays. In relation to cinema for education, there was a discussion on the possible production of a Netflix counterpart, which will be referred to as Uplix. We shall look forward to that. The 6th program is blending of multimedia approach and Satoyama Satomi principles for building climate smart communities, with program leader Dr. Bagarinao, collaborating with program manager Dr. Serrano, project leaders Dr. Boat Jr., Dr. Abito, and Dr. Kimbo, together with 4 project staff members, 3 research assistants, and 1 student. Their accomplishments include partnerships with LGUs and HEIs, collaboration with faculty member of University of Plymouth in UK, technologies for program surveys, interactive videos, and training materials, program management, surveys of stakeholders, focus group discussions, and presentation in international congress. The last project is promoting sustainable tourism in the small islands in the Visayas. The project leader here is Dr. Fernandez Avila, who works with 7 project staff members, 4 faculty members, and 1 student. It was reported that small islands found difficult situation across sustainability goals due to 1, ineffective implementation of policies and regulations, 2, decision-making process in which residents, especially the poor and vulnerable, felt blindsided, 3, problems related to workload, just compensation, and work-life balance, 4, need for improvements in safety and security, tourism attractions, environmental integrity, accommodation, food, and cultural aspects. And 5, evidence of negative impacts to environment and host communities. Swap toss analysis were conducted, which showed strengths such as diverse attractions and enabling environments, opportunities such as big demands for East Asian countries, strong linkages to research institutions and other partner institutions, facilitating tourism development programs of the government, utilities, presence of work problems, and negative perception and implementation and decision-making of tourism strategies, threats such as tourist dissonance, competition, climate change and natural hazards, increasing number of fixers, and the COVID-19 pandemic. Hopefully, the lessons derived from the project will become useful in promoting tourism in other small, amazing islands across the country. We now move on to the project reports to be presented shortly. Let us be inspired and be motivated to undertake more research. Thank you.
Thank you, PLC, for the recap. Let us now welcome our chair for this session, a member of the EMC and a professor from the College of Public Health, UP Manila, Dr. Nina Gloriani.
Yes, thank you very much, Fatima, and good afternoon, everyone. I'm really very happy, especially with yesterday's presentations. Of course, we listened to so many outstanding research contributions, top-notch researchers, and today we are going to listen to more. So we have, for my session at least, we have 4 that we are looking forward to hearing from. So for the first presenter, we have Dr. Cecil Leia Bayaga, who will be speaking on the influences of maternal dietary intake and nutritional status on the microbiological and chemical compositions of breast milk from selected lactating Filipino women at 0 to 4 months postpartum. It will be very interesting to find out how the mothers, ito yung mga dietary intake nila and their nutritional status will affect the quality of their breast milk especially. No? So we look forward to that. So without further ado, may I present Dr. Cecil Leia Bayaga. The floor is yours.
To Assistant Vice President Dr. Carla Di Malanta, Distinguished Professor Emeriti of the University, faculty members, fellow EIDR Cycle 8 recipients, students, guests, ladies and gentlemen, isang magandang hapon po sa inyong lahat. On behalf of our research team, I will share with you our research program entitled Influences of Maternal Dietary Intake and Nutritional Status on the Microbiological and Chemical Compositions of Breast Milk from selected Filipino women at 0 to 4 months postpartum. Our program is composed of 2 projects. The first one focused on the development and evaluation of a food frequency questionnaire that assessed qualitatively the prebiotic and probiotic intake of adult Filipino women. The second one concentrated on determining the microbiological, physicochemical, and chemical compositions of human milk obtained from urban living Filipino women at 0 to 4 months postpartum. Both of these projects were stationed at the College of Home Economics in UP Diliman. Originally, our program leader was Dr. Alonso Gabriel. With his untimely demise in 2020, I took over his role. The project staff, together with the program leaders, were responsible in collecting and analyzing all the data for the two projects mentioned. All project staff are either registered nutritionists, dietitians, or food technologists. In the process of collecting and analyzing data, the program was able to mentor CHE faculty and graduate students, particularly in advanced food microbiology and sterilization techniques. In my case, this research program was very instrumental to complete my dissertation under the PhD Nutrition degree program. To provide a brief background about our research program, the Philippines has an opportunity to improve child and infant mortality rates through concrete strategies to address the country's food security concerns. The MDG Achievement Fund Evaluation Report in 2013 entitled Ensuring Food Security and Nutrition for Children 0 to 24 months in the Philippines states that three outcomes were used in the evaluation. And one of these three outcomes is increased breastfeeding in the selected areas by at least 20% annually. In 2015, around 2.8 million Filipino children under 2 years of age were undernourished due to suboptimal breastfeeding practices. As we all know, breastfeeding is critical for the achievement of many of the Sustainable Development Goals. It improves nutrition, prevents child mortality, decreases the risk of non-communicable diseases, and supports cognitive development and education. As mentioned earlier, the program is on pre- and probiotics. To summarize the potential roles of probiotic bacteria for the infant, these are contributing to the reduction of the incidence and severity of infections in the breastfed infant by increasing mucus production and reducing intestinal permeability. And last, it also contributes to infant digestion through the breakdown of sugars and protein, which improve the intestinal habit, increase fecal moisture, also increasing stool frequency and volume. Why is there then a need to look into the chemical and microbiological quality of breast milk? First, if the chemical composition and microbiology of breast milk are indeed influenced by the maternal diet, then lactating Filipino women may be given more appropriate dietary counseling because breast milk is the sole source of nutrients and contributes to the microflora and health status of the infant. Problems on child mortality, stunting, and wasting shall be addressed at an early stage. Second, milk banks may benefit from the results of this program by encouraging more mothers to provide breast milk, especially to neonates with low birth weight. The stage of lactation and other maternal factors such as BMI, which is a reflection of the maternal diet, and regularity of consuming prebiotic or probiotic containing food items may eventually be included in the set of screening criteria for milk donors, as these factors may have an effect on breast milk quality. And then the last, the data to be generated may be used in planning and modifying nutritional recommendations for the lactating Filipino women in the future. Information as to whether breastfeeding women are consuming diets adequate to support the ideal breast milk composition must always be made available and monitored regularly. These are the general and specific objectives of the research program. Basically, we aim to establish the relation of maternal diet and other factors, such as age and BMI, among others, on the chemical and microbiological quality of breast milk in the early stages of lactation. We were able to accomplish this objective by, first, developing a validated dietary tool that assesses the pre and probiotic food intakes of Filipino women. Second, we measured the chemical components in the human milk, which included proximate composition, pH, total soluble solids, and human milk oligosaccharides. And then the third, we enumerated culturable microorganisms, which were total plate count, Staphylococcus aureus, lactic acid bacteria, bifidobacteria, total coliform, and E. coli. Our team is proud to share that the journal article containing the methods and results of project number 1 was a recipient of the 2020 Outstanding Scientific Paper Award by NAST. Our team was also able to acquire a certificate of copyright registration for the dietary tool developed to assess the pre and probiotic intake of urban living, low to middle income Filipino women. The developed FFQ consisted of 39 food items classified into 13 food groups. The frequency of consumption was described as daily, weekly, monthly, and never. Upon testing for reliability, 38 of the 39 food items had matched responses. Reliability was tested by having a set of participants answer the same FFQ in 2 separate occasions within the same month. Through this program, we were able to upgrade the Laboratory of Food Microbiology and Hygiene and create the Breast Milk Research Laboratory, both housed in the College of Home Economics. The acquisition of the ultra-low freezer has been beneficial to graduate students in food science and nutrition. Now, we have a proper storage place for our biological and food samples we use in our research. Now, aside from the development of a reliable and validated food frequency questionnaire and purchase of laboratory equipment, the results of the program are as follows. The chemical and microbiological content of human milk differ across lactation stages and are influenced by maternal characteristics, namely maternal diet, age, and body mass index or BMI. Among these 3 factors, maternal diet influenced both the chemical and microbiological contents of human milk. And then the last, our study or our research program was able to demonstrate that certain nutrients in the diet and the regularity of consuming selected prebiotic or probiotic-containing food items can affect or change human milk composition. Our team is pleased to share that the results of the research program have been published in reputable local and international journals. Finally, this research program was able to address 3 SDGs. One of the strategies adopted by the Philippine government to address child mortality, stunting, and wasting problems is to promote breastfeeding. Given the considerable health benefits it confers, human milk is universally considered the optimal source of nutrition for almost all infants. On top of this, breastfeeding also has benefits to the mother and to the family. And last, in our study, we found out that indeed maternal diet can influence milk composition. In some of the milk analyzed, the presence of harmful microorganisms such as coliform and E. coli were detected, and the possible sources of contamination may come from unclean water. Therefore, availability of potable water benefits the entire population. This is a health need that local government units has to address. For this presentation, these are the references that we have used. Dito po nagtatapos ang aming pagbabahagi. Maraming salamat po.
Okay. Thank you very much, Dr. Cecil, and that was great presentation. Congratulations to your entire team. This is something that we really need, no? The information to actually improve further yung mga health status ng ating infants especially, and of course the lactating mothers. So, I don't know if we have questions, but maybe I can actually ask. After you have done all of this, how will you proceed to, maybe educate or actually advocate for a change in the diet and, well, generally yung health status medyo mahirap, but the diet especially, how do you propose to do that?
Okay. Thank you, Dr. Gloriani, for that question. During the collection of the milk samples, we were in touch with the Makati Nutrition Office, and we would like to continue the partnership with Makati Nutrition Office. So we would like to start with them. So we have shared the results with the Nutrition Office, and hopefully during this the coming months or in the coming year, we would be able to draft a program or a nutrition education program to disseminate the information to the different barangay health centers. Kasi iba po ang pagpapackage ng nutrition information para sa mga barangay health centers. So, yun po yung kasunod na po na aming patutunguhan po. Pilot po muna sana with Makati, and if very successful, then we can open what we did to them with other cities, particularly siguro din po sa Taguig at sa Mandaluyong, kasi ito rin po yung dalawang cities, at dito rin po sa Quezon City, ito po yung tatlong cities na pinuntahan namin for the sample collection.
Yes. Thank you very much. And I'm happy to also note, no, you have upgraded your laboratory. You know how important it is.
Yes.
UP is actually supporting all of this now. Yung way back do sa amin, we had to really parang cry for this. Pero ngayon, I'm seeing that most of the laboratories are fully and well equipped. Oo. I have here yung congratulations and thank you from VPAAG Cynthia Bautista to your team, Dr. Bayaga. So, meron pa po
Thank you po,
mga
VP.
questions? Yung iba dito. But I'm happy that you can actually cascade it down. Actually, ano pa ba yan? Kailangan kasi all of our results sa research should benefit the communities, especially this type of project. So, congratulations again. And yung sa Makati will just be a start.
Yes,
Breastfeeding is a very important public health program sa atin. So you're in the right parang direction, no? Thank you very much.
you po.
UP is actually doing all of this. So okay, if there are no other questions, thank you very much again, Dr. Cecil.
Thank you po.
We move on to our next presenter. So pero kung may mga questions pa, you can actually I have been instructed to ask our presenters to put their email addresses kung may mga questions dito sa chat box daw, and all the other presenters, just to help our participants, yung ating audience today. So for our next presentation, we have Dr. Lilibet Salvador Reyes. She will be speaking on targeting, microbiology ako no? Targeting microbial pathogens using specialized strategies and bioactive molecules of shipworm symbionts. I will have to admit na hindi ko masyadong kilala yung symbionts. Sabi ko, sino ba itong mga shipworm na to? Nag-google ako, and I found it very interesting that because I'm in microbiology, meron silang symbiosis, and you can actually draw a lot of parang use applications for all of them. So, salamat, no? This will be a very important presentation as well. So, ito yung tinatawag na invertebrate ba? And bacterial symbiosis for some applications. At pwede rin ito sa sa iba-ibang mga applications, not just health and others. So, without further ado again, we have we're Presenting here, Dr. Lilibet Salvador Reyes. Take it away na.
Good afternoon to everyone. I'm happy to share with you the accomplishments of our EADR-funded project entitled Targeting Microbial Pathogens Using Specialized Strategies and Bioactive Molecules of Shipworm Symbionts, or Project Tamilok for short. This program consists of 4 projects implemented by the Institute of Biology, Institute of Chemistry and Marine Science Institute, the College of Science, University of the Philippines, Diliman. Project 1 is led by Dr. Joyce Ibana and focuses on the interactions between shipworm microbial symbionts and pathogens. Project 2 is focused on the global metabolite profiling of shipworms and led by Dr. Hias Hunyo. Projects 3 and 4 are geared towards purification of bioactive small molecule compounds and enzymes from shipworm microbial symbionts. Dr. Isadora Yu leads Project 4, while I am in charge of the overall program supervision and activities under Project 3. If you're headed to Palawan or Aklan over the holidays, you'll probably be offered to try shipworms. These are locally known as tamilok. Don't be afraid to try this. These are not worms, but they are marine bivalve mollusks related to mussels, clams, and scallops. So they should taste like clams after all. What you will likely be offered would be Pterido navalis, shown here in this picture. It's one species of shipworms that thrive in submerged and decaying wood from mangroves. But actually, shipworms are more fun in the Philippines. The seminal work of the Philippine Molus Symbiont ICBG showed significant biodiversity of shipworm species in the Philippines, with several new species discovered in the last few years. For example, Tamilocus mabinia from Mabini, Batangas is a new species of wood-boring shipworm characterized by an extremely long digestive system. The unicorn of the shipworms, the giant Cuphus polythalamius, found in Sultan Kudarat, relies heavily on hydrogen sulfide for nutrition. Then, there is the rock-boring shipworm, Lithurido abatanica from Bohol, that is unlike any other shipworms. It lives in a freshwater environment and bores through rocks. The digestive strategy of Lithurido abatanica is currently unknown, and this is something that we can all look forward to. Despite the different lifestyle by shipworms, these organisms rely heavily on their microbial symbionts for critical functions. For wood-boring shipworms, the cecum is the site of wood digestion, while special cells in the gills houses microorganisms that are needed for critical functions such as wood digestion and nitrogen fixation. Surprisingly, the cecum of shipworms are largely devoid of microorganisms, suggesting a mechanism employed by shipworms to structure the microbial community. Metagenomic and culture-dependent techniques indicated that shipworms with different lifestyle would have variable microbial population. In the wood-boring shipworms, Pteridinibacter turnerae is the dominant microorganism. This gamma proteobacterium devotes a significant proportion of its genome for the production of natural products. Therefore, shipworms would have a symbiotic relationship with the microorganisms in their gills, relying on these microbial symbionts for enzymes needed for critical functions such as carbohydrate degradation and nitrogen fixation. At the same time, the microbial symbionts in the gills of shipworms would also interact with microbial pathogens they encounter from the environment. Microbial pathogens are inhibited in part by small molecule compounds produced by shipworm microbial symbionts. The strategy employed by shipworms in their microbial symbionts using small molecule compounds and enzymes has been very much successful, and these methods have been evolutionarily optimized for millions of years. Hence, we are interested in tapping small molecule compounds and enzymes produced by shipworm microbial symbionts for biotechnological and biomedical applications. This program aims to develop alternative potential therapeutics to combat drug-resistant infections from the small molecule compounds and enzymes produced by shipworm microbial symbionts. We were very fortunate to have been able to collect shipworm specimens in Balayan Bay in Mabini, Batangas a month before the start of the lockdown in 2020. A total of 13 shipworm specimens were collected and extracted from wood. These were dissected to obtain 41 host tissues in all. The tissue samples were extracted on-site, and the resulting homogenates were plated to give 205 microbial plates. After this, the morphologically distinct colonies were purified by repeated streaking, and these are now cryopreserved for long-term storage. We were able to purify a total of 267 microbial isolates from the different media we use. Because of the limitations from the COVID-19 quarantine measures, we decided to focus on microorganisms that were collected or purified using the shipworm basal medium. These microorganisms share similar morphology to Pteridinibacter turnerae, and we also observe clearing zones in the agar plate. The clearing zone is indicative of cellulolytic activity. We obtained 35 microbial isolates from this collection and combined this with 5 reference strains from the PMS-ICBG collection for our use in the next round of activities. The 40 microorganisms were subjected to liquid fermentation and extracted with diion HB20. The extracts were subjected to biological activity assays, and the secondary metabolites were profiled by mass spectrometric-based method. Based on the bioactivity and the chemical profiles, we prioritized several of these microbial isolates for large-scale extraction and purification of carbohydrate-degrading enzymes and small molecules with antimicrobial activity. In the succeeding slides, I will share with you the results of our antimicrobial screening, global metabolite profiling, as well as purification of small molecule compounds and enzymes from shipworm microbial symbionts. First, let's take a look at the antimicrobial activity of these extracts. The extracts were tested for their antimicrobial activity against the microbial pathogens Staphylococcus aureus, methicillin-resistant Staphylococcus aureus, and Bacillus subtilis using the microbroth dilution assay. This heat map shows the activity of the extracts against the microbial pathogen. Red would be indicative of close to 100% inhibition of the microbial pathogens, while yellow would indicate close to zero inhibition of the microbial pathogen. Hence, extracts with a yellow color would be inactive. We can see here that approximately 50% of the extract inhibited Staphylococcus aureus at a concentration of 50 microgram per ml. A concentration-dependent inhibition of Staphylococcus aureus is also observed, with fewer extract inhibiting this pathogen at a low concentration of 0.5 microgram per ml. These extracts would also have different spectrum of selectivity. Only one particular extract, labeled E9, showed a broad-spectrum antimicrobial activity. This extract inhibited not just Staphylococcus, but also the methicillin-resistant Staphylococcus aureus, as well as Bacillus subtilis. We also tested the extracts of non-cellulolytic microorganisms from shipworms for antimicrobial activity. Unlike the extracts from cellulolytic isolates or those morphologically similar to Pteridinibacter turnerae, those microorganisms that were non-cellulolytic were largely devoid of any antimicrobial activity. As you can see from this heat map, except for one extract labeled E64, everything else were devoid of any significant activity. Therefore, extracts from cellulolytic and non-cellulolytic isolates from shipworms would have also very different antimicrobial activity. To obtain insights into the chemical diversity of the extracts from the cellulolytic isolates, liquid chromatography coupled to high-resolution mass spectrometric analysis was carried out. This was analyzed using the Global Natural Product Social Molecular Networking Tool, or GNPS, and the results are visualized using Cytoscape. GNPS is the Facebook of molecules. You can basically see the relationship between molecules, which are closely related to one another based on their chemical structure. What we see here would be a representative molecular networks of compounds from the shipworm microbial isolates. This is an example of a molecular network, and a molecular network would consist of structurally related compounds. So this is one family of structurally related molecules in the extracts. The molecular network consists of nodes, which would be equivalent to one mass spectrum, approximately equal to one compound. We can see here from the molecular network a diverse family of molecules produced by shipworm microbial symbionts, corroborating with the genomic analysis indicating high biosynthetic potential of these microorganisms. When we compare the molecules produced by the reference strain Pteridinibacter ternary T7901 with those from microorganisms collected from shipworms in Mabini, Batangas, we can see similarities and differences. The red dots here indicate compounds produced by T7901, which is the reference strain, while the blue dots are from the isolates from Mabini, Batangas. So we can see here that like T7901, the microbial isolates from Mabini, Batangas would also produce the antibiotic partalones. In contrast, if we look at other molecular clusters, we can see now distinct and more diverse chemistry from the microbial isolates from Mabini, Batangas. You can see here that in this molecular network, we only have a few molecules produced by T7901 in contrast with a more diverse molecules produced by the isolates from Mabini, Batangas. From the results of the bioactivity and chemical profiling, we prioritize 5 microbial isolates for large-scale fermentation. We employed the bioactivity and chemistry-guided purification. Repeated cycles of column chromatography were done, and in the process, the resulting fractions were tested for bioactivity, and the chemistry was replicated using mass spectrometry. We then undertake further compound characterization using nuclear magnetic resonance spectroscopy. Here you can see a representative example of a proton NMR of one of our priority compounds showing characteristic chemical signatures for modified oxylipins. We have so far purified modified oxylipins with biofilm inhibiting activity as well as peptide-based molecules, and we hope to share this with you in the future. To obtain carbohydrate-degrading enzymes, we mine the genomes of 9 cellulolytic shipworm isolates, and we prioritize genes with potential enzymatic activity against extracellular polymeric substances in biofilms. So from a total of 813 total genes, 77 candidate genes were targeted for gene amplification, and we selected based on the substrate specificity and novelty. We wanted to target enzymes with low sequence homology, or these are unique protein sequences, and those without a 3D protein structure. After this, we carried out PCR amplification of the target genes, cloning, verified the sequences, and these were transformed into E. coli expression host, and protein expression and purification were carried out. We have successfully expressed and purified an endocylinase with in vitro activity. We have 2 more enzymes that have been expressed and are now ready for purification, and we hope to share with you the results in a future discussion. In summary, what I want to leave you with would be diversity. Project Tamilok has so far shown diversity of the microbial isolates. There's also organismal diversity of shipworms in the Philippines, We have a lot of chemical diversity in the extracts of shipworm microbial symbionts that translate to functional diversity, with the extracts showing a wide range of biological activity and enzymatic activity. Finally, we are grateful to our collaborators, the Philippine Molus Symbiont ICBG. This is led by Professor Marco Haygood of the University of Utah and Professor Emeritus Gisela P. Concepcion. We are thankful to the Office of the Vice President for Academic Affairs for the funding for this project, as well as the College of Science and our home institutes for their support, especially during the lockdown of 2020. We are grateful for the assistance of the Bureau of Fisheries and Aquatic Resources, as well as the Municipality of Mabini, Batangas, for allowing us to conduct the sampling of shipworms in their locality. And to you all, thank you for your kind attention and have a good day.
Wow, thank you very much, Dr. Lilibeth, no? I'm really awed with what all of these researchers could do. So yung richness of our flora and fauna, and you were saying yung organisms, yung diversity ng microorganisms, yung organism level, and yung chemical ano nila. So there's really a lot of potential in this. Especially when you mentioned about biofilm formation. Natutuwa ako kasi retired na ako, no? But I had some students working on biofilm formation, and we were testing many agents, and I'm very sure your product will be able to help us, especially in the medical profession. Ano ba yung mga biofilms? Diba ito yung mga tao, yung mga patients nakasuot ng mga catheters, yung doon pumupunta yung nagde-develop yung biofilm, and a source like this from the Philippine seas will be very helpful. So we have here congratulations again from Dr. VPAA Cynthia Bautista to you for very prolific team, productive, and we look forward to yung actual na biological activity nitong mga to, and of course we can always yung inyong as early as this time you should be able to already network or connect with potential users. So
Yes.
what else can you actually
So we've been also in contact now with aquaculture communities doing the farming for shipworms, and we hope to partner with them. Because again, this has also now huge potential, not just as for biomedical applications, but shipworm itself is now promoted as a superfood. So right now, it's like a tourist offering, right? But because of its potential, yung fast growth and all that, it's now being promoted as the food for the future. So there will be a lot of work on shipworms for this. And then I think Professor Emeritus Mendoza has a question regarding the candidate bioactive molecules that will need follow-up studies. So we actually have these are modified oxalipins, so fatty acid molecules, and we determine a structure, and then we're doing structure activity relationship, so looking at which parts of the molecules would now have an impact on the biofilm activity, and we're able to image the effects of these inhibitors on your biofilms. So we use the SEM to image them, and we see an AFM also to see their effects.
So that's really great. So Ma'am May, are you fine with that answer? There's really a lot more to do with so much.
Oh yeah, this is just the beginning.
We're very
Lilibet, this is just the beginning.
Yes,
no?
and
You can see so many things that need to be followed up, and you will need, of course, funding support and more students and collaborators. So really congratulations, no? Hindi ganito yung nabasa kong the recent report na nabasa ko, hindi ganito. Congratulations. Maganda rin ang presentation. Congrats to the team.
Yes.
Thank you, ma'am.
Thank you.
And I'm happy to
The
share.
VPAA, Cynthia.
We now have three proposals that will that were submitted. They're already submitted on the follow-up works for shipworms, and so we're happy with that. And I guess that's one effect of the pandemic. Also, we were only able to tackle just one site. If you've noticed, we just did Mabini because even before that, we couldn't get the prior informed consent from the communities because the borders were already locked down, even before the March 2020 lockdown. And then at the same time, we only focus on a subset of the microorganism. So with our current collection, and they're still cryopreserved in the lab, anytime we can wake them up and work on them again.
Yeah.
We can continue the work. And so that would be the topic of three more upcoming proposals.
Wow, thank you very much. VPAA, Cynthia wants to...
No, I just wanted to say this is very heartening and congratulations. It's very important.
Thank you.
I guess PE May is here. The NAS division of Pagtanaw 2050 is really a vision for of an archipelagic nation and an economy that is based on the fact that we are an archipelagic nation. And we are reviving. We used to have an archipelagic studies program in the university where we had the marine at that time, Ed Gomez and the marine scientists, and then we also had the lawyers and also had the social scientists who were studying the rattles then, but that did not continue. So we are reviving that, I guess, to lay the groundwork for possibly a future network of a maritime university. Internal maritime university. I mean, virtual. It's not a university in campus, but it's a virtual connection. So this is, it's heartening that we have this, and we will be connecting this and other projects very soon.
Yes, thank you very much, VPAA, Cynthia. And there's really so much promise potential here. We look forward to brighter things for your group. Congratulations again, Dr. Lilibeth. And
Thank you very much. Thank you very much, VP Sinch, for your support. Thank you.
Okay, so we move on to our third presentation. They're somehow related, no? So now we have ito naman, prebiotic sugars, no? Parang mga pagkain yata ito, mga kinakain kanina yung Symbion. And then, of course, yung breast milk. And then, we now have this. For our third presenter, we have Dr. Cheryl Ariola, Associate Professor from the College of Arts and Sciences of UP Los Baños. She will be presenting their studies on structural and prebiotic studies of galacto and heterooligosaccharides formed by, you're familiar with lactobacillus, no? Pero ito lactobacillus delbrueckii, subspecies bulgaricus, DSM, and then on yung beta-galactosidase. I know the beta-galactosidase has many uses, and I look forward to actually hearing from you. So, Dr. Cheryl, the floor is yours.
Good afternoon, everyone. Today, I'm going to share with you the result of our research entitled Structural and Prebiotic Studies of Galacto and Heterooligosaccharides formed by Lactobacillus delbrueckii subspecies bulgaricus DSM 20081 beta-galactosidase. I am Cheryl Ariola from the Institute of Chemistry, UP Los Baños, and my collaborators include Dr. Francisco Elegado of Biotech UPLB, Dr. Dietmar Haltrick of Bukovina, Austria, and Dr. Carlito Librilla of UC Davis, California, USA. Enlisted here are the research assistants who work with me to achieve the project objectives, and several UPLB graduate and undergraduate students who were supported by this EIDR project. Our project aimed to characterize galacto-oligosaccharides synthesized by the enzyme beta-galactosidase, also known as lactase. It is characterized as hydrolase. This enzyme reaction requires two carboxylic groups, one to act as a catalytic nucleophile and the other one to act as an acid-base catalysis. During the initial stage of catalysis of this enzyme, where lactose is used as the substrate, glucose is released from the active site, and a covalent galactosyl enzyme complex is formed. The galactose from this complex is transferred to a galactosyl acceptor, which could be present in the reaction mixture. Water could be an acceptor, thus favoring the lactose hydrolysis, forming glucose and galactose. This reaction is commonly used in dairy industry to produce lactose-free or lactose-reduced milk products, and in cheese whey production to minimize environmental problems. In addition, uh, any carbohydrates present in the solution could act as galactosyl acceptor. It could be glucose or galactose, the hydrolysis product of lactose. It could be lactose forming galacto-oligosaccharides. Galacto-oligosaccharides are considered prebiotics. When we say prebiotics, these are non-digestible dietary components that pass through the colon and selectively stimulate the proliferation and/or activity of desirable microorganisms in situ, thus improving health. Other well-known prebiotics include inulin, fructooligosaccharides, and lactulose. In our digestive tract, the digestive enzymes slowly hydrolyze galacto-oligosaccharides and fermented by colonic bacteria, producing carbon dioxide, hydrogen gas, and short-chain fatty acids. Acetic acid, butyric acid, propionic acids are the most common short-chain fatty acids produced during carbohydrate fermentation. High level of these SCFAs is concomitant to decrease in luminal pH, which could result in the decrease of the population of Salmonella, Helobacteria, and other harmful bacteria. That could also lead in the increase in population of beneficial bacterial populations such as those lactobacilli and bifidobacteria. Thus, this could lead in the improvement of the general health of intestinal epithelial cells. In our previous study, we used different sources of beta-galactosidase, one from Bulgaricus, from Reuteri, and two from Bifidobacterium brevi. We used same condition in the production of galacto-oligosaccharides, that is, 205 gram per liter lactose as the initial substrate concentration. We added 1.5 unit enzyme per ml, and the lactose was previously dissolved in 50 millimolar sodium phosphate buffer pH 6.5. We also added 10 millimolar of magnesium because magnesium enhanced the stability of the enzyme. We found out that among these 4 beta-galactosidases, the one from Lactobacillus bulgaricus gave the highest total GOS yield of about 50%. We also found out that this enzyme could transfer galactosyl moiety to N-acetyl D-glucosamine to form N-acetyl glucosamine containing oligosaccharides, which could be beneficial to food and feed industry. Thus, our project objective is to develop analytical strategy to characterize GOS and GlucNAc-OS synthesized by beta-galactosidase from Lactobacillus bulgaricus. The specific objectives include to produce and purify GOS and GlucNAc-OS, separate the size-excluded oligosaccharides by HPLC, with a porous graphitized carbon column, elucidate the structure of oligosaccharides by LCMS, and assess the bifidogenic effect of the size-excluded oligosaccharides by bacterial growth experiments. For our methodology, we have the substrate. So the substrate depends on what product we want to have. If we want to have just GOS, then we use 600 millimolar lactose. If we want to produce glucnac-OS, then we add 600 millimolar of N-acetylglucosamine. We add a certain amount, 1.5 unit enzyme, and incubated for a certain period of time to produce the crude oligosaccharides. One drawback of enzymatic transgalactosylation is that the production of lactose hydrolysis products, glucose and galactose. And these monosaccharides have high caloric value, about 4 kilocalorie per gram, thus limiting in use in food and pharmaceutical industries. Additionally, these monosaccharides can contribute towards growth of lactic acid bacteria, which could hamper the true assessment of prebiotic properties of the galacto-oligosaccharides and N-acetyl-containing oligosaccharides. Thus, we did different optimization to remove these monosaccharides. We performed yeast fermentation, different types of uh, chromatography, so that we can remove the monosaccharides in our GOS mixture. And we found out that among these techniques that we have performed, gel filtration using Biogel P2 is the best one for our objective. We monitor, so we loaded our crude GOS on the column, and we monitor the elution by determining the total reducing sugar for each fraction. And we further evaluate the carbohydrate content in the fraction using TLC. Those fractions that do not contain monosaccharides were pooled and reduced with sodium borohydride for LCMS analysis. For bifidogenic effect, we use probiotic bacteria, Lactobacillus bulgaricus and Lactobacillus reuteri, to evaluate the effect of the GOS as carbon source in their growth rate. So here is the structural characterization of enzymatically synthesized oligosaccharides. Using QTOF MS, we were able to characterize the different degree of polymerization of the synthesized oligosaccharides. We have here the partially purified GOS and the partially purified glucnac-OS. Upon selection of the specific mass-to-charge ratio, we were able to identify 6 distinct isomers of trisaccharides, 7 distinct isomers of tetrasaccharides, and 5 distinct isomers of pentasaccharides. More so, when we have lactose and acetylglucosamine as the substrates, we have 3 distinct isomers of gal-glucnac, 7 distinct isomers of gal-gal-glucnac, and 3 distinct isomers of gal-gal-gal-glucnac. Here is the tentative characterization of the reducing GOS. So we have here the lactose, and the enzyme showed intramolecular transgalactosylation activity. It was able to synthesize the regioisomer of lactose, which is 3-galactosyl glucose and 6 galactosyl glucose, which is allolactose. This disaccharide was elongated further by 1,3 linkage and 1,6 linkage. Furthermore, this allolactose was elongated by 1,6 linkage and 1,3 linkage. The lactose served as the galactosyl acceptor, thus forming further elongated by 1,3 linkage and further elongated by another 1,3 linkage, forming tetrasaccharides. In this side, we also have here the lactose is elongated by beta 1,6 linkage and further elongated by another 1,6 linkage and another 1,3 linkage. We can also see from here that the enzyme was able to synthesize galactobios. That is 3 galactobios and 6 galactobios. We were not able to detect non-reducing isomers, primarily because these non-reducing isomers could not be reduced by sodium borohydride. We also performed a fermentation study to evaluate the effect of the partially purified GOS on the growth of non-probiotic bacteria Lactobacillus reuteri and Lactobacillus bulgaricus. We used 0.1% glucose as the control carbon source. After 8 hours of incubation, we found out that higher growth rate of Lactobacillus bulgaricus and Lactobacillus reuteri was observed when glucose was used as the carbon source. The lower observed growth in GOS and lactose can be due to the presence of oligosaccharides with high degree of polymerization, as these microorganisms need to hydrolyze the oligosaccharides prior to consumption. It's also worth noting that the prebiotic effects of GOS affect monomer composition, type of glycosidic linkage, and degree of polymerization of GOS. So we also try to enhance quality of different food products by using either the enzyme or the partially purified GOS. Lactose intolerance is one of the problems here in Southeast Asia, and dairy industries are trying to produce lactose-free products or lactose-reduced products. So what we did is we used cow's milk, carabao's milk, and goat's milk as the substrate, and we add enzyme and monitor if this enzyme could hydrolyze the lactose. We found out that indeed this enzyme could hydrolyze lactose and at the same time synthesize the galacto-oligosaccharides. So this suggests that this enzyme could minimize lactose concentration in dairy products, which could benefit those lactose intolerance. More so, we have here we fortified sugar cookies with galacto-oligosaccharides. So we have a control, 0% GOS, 10% GOS, 5% GOS, 10% GOS, and 15% GOS. We found out that A had the highest overall acceptability and C has the lowest overall acceptability because this C had a darker color and higher degree of spreading. So if in case we want to fortify our sugar cookies with GOS, we have to make sure that there is a balance between the amount of GOS and the overall acceptability of the target population. So showing here are the references that I used in this study, and thank you everyone for listening to this presentation.
Wow, thank you very much, Dr. Sherry. No? Nakakatuwa naman talaga. All of this will benefit our people. Toto nung lactose intolerance, that's a big problem here, no? So, I don't know if we have questions right now. Wala pa. Meron tayo dito from VPAA, Cynthia Bautista again. Congratulations daw to you. Thank you to Dr. Cheryl and your team. Marami talaga, sabi ni ma'am, significant implications for the health of our communities. Ang tanong ko, ako na lang muna magtanong, sinong local milk manufacturer ang may lactose-free milk? Because what we buy, I buy for my auntie, no? Puro parang mga imported to. So that will be part of your, sino yung bebentahan ninyo yan? Yung
I really don't know if we have commercially available, locally produced lactose-free milk products. For our study, what we use are those products from Philippine Carbo Center and from our UPLB-D3. So they are not lactose free, but maybe with the collaboration of D3, we can produce lactose free products for lactose intolerant.
Yes, thank you. Malaki ang market niya na baka maging mura pa because it's going to be local. So do we have question, Ma'am May?
Yes, thank you very much. Congratulations, Cheryl. Ang galing.
Thank you, ma'am.
Bakit magagaling ang presenters natin ngayon na women power to yata, pati. Ang question ko, paano magagamit na para sa commerce? Yung, actually, parang katulad nung kay Prof. Nina, no? Yung bang magpo-produce ng oligosaccharides, galactose, mga oligosaccharides for the processors, no? Yung tamang or one of them is to really produce the milk, the lactose-free milk. Ano nakikita nyo diyan pagkakakitaan ng pera? BPC?
It's a big market, ma'am, because we Filipinos are basically lactose intolerant. When 90% of the population in Southeast Asia are lactose intolerant. The only problem is that we don't have much milk source. We import milk from Australia, New Zealand, and European countries. Konti lang po kasi yung source ng milk natin. So siguro, kung tayo ay makakapag-produce pa ng more milk, then we can enhance that product pa po. What we are doing actually in the lab right now is another class of prebiotics. Kasi po, konti lang po yung source talaga natin ng gatas. What we have now ay mga manan. So these are mono-oligosaccharides. This is a new class of prebiotics. And medyo marami na po kaming salamat sa AIDR because basically the pieces of equipment that we used in this project was used for the mono-oligosaccharide production. So doon po kami ngayon sa production ng mono-oligosaccharide, another class of prebiotics. And this time we are using halimbawa po coconut milk naman po for production of prebiotics. Doon po kami medyo nag-shift kasi konti lang talaga yung source ng milk na meron tayo.
So thank you very much, Dr. Cheryl, Ma'am May. Thank you. So any other questions or meron pa tayong 1 minute? So tanungin si Dr. Cheryl, saan pa magagamit? Oligos. Alam nyo, mahirap sa akin yung mga chemistry na yan eh. Mas protein ako kaysa sa mga sugars. Masama din sa akin ang sugar. Pero I look forward to the lactose-free milk that you may produce.
Hindi, ma'am, yung GOS ngayon, inilalagay na nila sa infant formula because they want to mimic the human milk oligosaccharides.
Oh, they're good.
Yeah. Meron na po talaga, ni-incorporate na po nila. Marami na akong nakitang products dito sa Pilipinas na supplemented by GOS.
So, paano mo gagawin yon pagka gusto mong mag-produce ng the right kinds of oligosaccharides? No? So, papunta na nga yan sa commercialization, Cheryl.
So, dapat ma'am, alam mo kung kailan ka mag-stop ng reaction and you just have to remove the monosaccharides. Kasi ma'am, ang taas ng monosaccharide content kapag yung crude GOS. So, you have to do certain filtration so that you will have more or less pure GOS that can be incorporated to other food products po.
Marami pang optimization ba? Ganun ba yun?
Well, the biochemist will be able to address that problem. Cheryl.
Yes, ma'am.
with additional support from UP system, ano po, BPA. More, kailangan natin lahat yan. So thank you very much, Dr. Sherry Lariola, for that wonderful talk and very promising as always, no? Sana yumaman na tayong lahat, kayong lahat sa UP. We have very top-notch na mga scientists na we really have to take care of you. So thank you. So dito we move on to your team also. No? We move on to our last, definitely of course not the least for this session. Ito po yung tungkol naman sa fish o kakainin na naman. So, I am very happy to present to you si Dr. Ricardo Babaran. Ito actually, I had a chance of actually reading through their many accomplishments. So, looking at the applications of molecular genomics and otoliths for fisheries management programs. So, gone are the days when we just fish and use the fish. Ngayon po, scientific na the way we actually characterize. Nakakatuwa, may museum pa si na Dr. Babaran. So without further ado then, so Dr. Ricardo Babaran, please, we look forward to hearing your presentation. Thank you po.
Good afternoon. This is our project title, Applications of Molecular Genetics and Otoliths as Tools for Fisheries Management. The fisheries of the Philippines need management, and the reasons for this management is that the Philippines is a biodiversity hotspot. Our country's resources are progressively declining, but are not yet fully studied. Our main resource, tuna, which is about the $350 million industry, has long been in need of science-based interventions. The study of reproductive biology of fish, one of the key requirements for aquaculture, is so limited to a few species. Our fishermen still remain poor, and if we address all these, we're addressing Agenda 1 of SDG, Agenda 2 of SDG, and number 8 of SDG. We combined together 4 projects for this: DNA barcoding of fishes of Western Visayas, development microsatellites and NSP markers for selected populations. We want to study the population structure and connectivity of yellowfin tuna. And then we also would like to determine the age, growth, and reproduction of commercially important carangid species. These are our the members of our project team. We supported over 10 BS and MS students of UPV. Some of them became our staff eventually. Some of them became members of SUC when they got out of the university. For Project 1, Panay and Guimaras Island are rich in marine fishes, including rare and understudied species. And we knew this because of a previous study that we participated in 2012-2016. And there are still a lot of studies that fish that need to be identified. Aside from that, there's little genetic data that has been published for Western Visayan fishes. So for Project 1, the objective is to document marine fish species diversity in Western Visayas and to contribute towards DNA barcode reference libraries and expand the voucher specimen collection that we have here at UPV. The project collected 2,000 samples representing 57 families. 304 species were identified, mostly bony fishes. And we have sequenced so far 185 species. The most represented groups are the grouper group, the cardinal fishes, the surgeon fishes. But we have a good representation of also of other species, which are shown here on the left. After assessing our performance during the past 2 years, we noticed a tapering of the number of species that are identified through the years. So it seems that from the sites that we focused on, our collections are more or less complete. Highlights for Project 1, we upgraded the UPV Museum of Natural Science Research Laboratory. So we identified 253 species from 304 collections. the IDs of 50 on taxa are still uncertain. This is nice because this is something that we can focus on for our publications. But we would like to focus on families that are unidentified, like flying fishes, parrot fishes. We would like to also focus on undescribed species, like lizard fish. And then we also would like to see species that have high phenotypic polymorphism. Through the project, we also try to match what EIDR gave to the project by coming up with a database for our museum right here. This is a slide showing the special focus on flying fish. These are juvenile fishes. There are two groups, and turns out that the pictures that are given in the database, Global Gene Bank, are not correct. There are misidentifications also noted for this group of species. And then for the impacts of this project, our members attended 2 conferences. Our group members from this project are now being popular. They are invited to train their peers in other universities. We supported 2 graduate students, and we're in the process of preparing a book called The Checklist of and DNA Barcodes of Marine Fishes from Western Visayas. This is an output from previous work. A continuation of output from previous work. Carangids represent a large group of species that are captured by Hurricane, which is the most dominant commercial fishery in the Philippines since the 1970s. A species identification is the key is a key requirement for fisheries management, and population genetics of dominant small pelagic fish species like scads that are shown on the right is not yet well studied. This work is a continuation of what we did in Japan with Japanese scientists since 2012. For this project, we want to discover and develop genetic markers for fish population genetics. And we want to use these markers to investigate 2 particular species, the longgong, the Captus macrosoma, and the Selarcum neophthalmus, matang-baka. The highlights of this project, highlights of accomplishments for this project. 2 is the generation of high-quality de novo genome assemblies for Decapterus and Selarcum neophthalmus. We assembled the complete mitochondrial genome of these 2 species. We made already initial investigations of the population genetic structure of Selarcum neophthalmus using mitochondrial CO1 marker. And as a continuation of the previous work, this project and the next project collaborated to barcode 1 Decapter species, and we are about to have published 1 paper for that. This is the Decapterus macrosoma. The draft assembly. Draft assembly yielded 724 mega base pairs genome. And our initial results show that this is 97.8 complete. This is a similar result for Thalamus. The genome is slightly less, 576 megabase pairs, but the work is almost also complete, 98.5%. We have a complete assembly of the mitochondrial genome for both species, and we can use this now to make an initial assessment of the distribution, population genetics of both species. So we did that. We went to 7 sites in the Philippines, and our initial results reveal that there is heavy mixing for both species if we just make use of the CO1 marker. So what we intend to do is use more sensitive, finer scale population structuring, more sensitive markers, SNPs, for example, to determine finer scale population structuring for both species. So this is the highlight for this project for now. We continued, as I've said, from previous work. we have already submitted a paper for this one, but just to highlight this particular fish, our result gives the first report of molecular barcodes for this Smith-Vanisi species. The tree that is shown in the left now reveals how this fish relates to other Decapterus species. And then the red fin that you see for this fish seems to be meaningless because the Decapterus species seem to be mixed with other non-red finned species. If you just see this structure. Okay. Project 3. So the focus of this is a continuation of our previous work. Since the 1970s, we have been producing tuna in large quantities until 2005. In 2005, we were producing about 500,000 metric tons. And for this reason, the Philippines was deemed to be a very effective fishing country. And our fishers in the West and Central Pacific Ocean were constrained to just operate within 2 months. As a result, a lot of our fishers were sent home. Many companies went bankrupt. And because of this, it's important to look at the geopolitics of resource use. I think we want to look at the connectivity of the resources in the Pacific to what we have in the Philippines so that we can probably leverage what we have to how we can access resources in the Pacific. This work is a continuation of what I did, for example, in Kagoshima University, and what we also did in collaboration with others with Wageningen University. So for this particular project, we want to establish a otolith research laboratory at UPV. We want to develop protocols and conduct training on otolith chemistry and other otolith-based studies. We want to evaluate the feasibility of discriminating the young tuna species that are located in our country so that we can use them to investigate the origin and movement of the large species in relation to ours. Our project was able to this is project 3 supposedly. We already established the laboratory. We were able to collect our samples needed except for one area. And we were able to, in the meantime, while we're collecting, completing our samples, we imaged most of our samples in preparation for paper that we're producing. And then we already made arrangements to commence our chemical analysis for our otolith samples. For project 3, we participated in 2 conferences. Our project team members participated in 2 conferences. 2 training workshops were conducted. We established a link with the Balik scientist, Dr. Gerald Duancas, and we're going to host another one, Dr. Salvador Taron, this June. So one publication is now being prepared. We are preparing an atlas of otolith fishes of Panay. This is a collaboration activity funded with also with by UPV. And then we are anticipating, we submitted, what we did was we leveraged this proposal, and we submitted the 2 proposals to the UST. So those are prepared and submitted, and then we're anticipating new international partnerships because of these proposals. For Project 4, the context of Project 4 is that in Japan, for example, fish like the one on the right have been cultured in Japan. There are similar species in the Philippines, and the wild populations that we have are overexploited. There's no national policy. Our national policy for fisheries is more tilted towards aquaculture, and our studies are more several species like milkfish and tilapia we believe are overstudied now. Many marine fish species with great potential for aquaculture are similarly not getting enough attention. New conservation management paradigms are one thing, and policies towards integration of subsectors of fisheries is needed. Some of the basic studies that we are doing for this project were done in the past, but much more is needed. So we want to the laboratory that we want to establish for Project 3 is also a target for this project. We want to catalog the otoliths of commercial species, mainly for their age. And also we want to gather more data of the age, growth, and reproduction of commercially important carangid species in the island of Panay. So we collected samples, enough, I think, for us to publish our results for this particular project. In the process of doing that, we strengthened our project team in terms of fish population dynamics, reproduction, aging studies, and we supported 4 BS fishery students, undergrad, mentored by our project team members. And then we also prepared a fish otolith aging protocol for the captures and cellar species. This we intend to use to collaborate with others later. Outputs for this project, a significant outputs for this project. So we attended 2 members of our project team attended the conference. Training is basically internal, but this is for strengthening purposes. We are anticipating the support the publication of 2 papers. One is already submitted to Asian Fisheries Science. Another is going to be submitted to Fisheries Research. And the data, information that are gathered through this project will be used for fisheries policies towards management, conservation of Now, it is important to emphasize that the program is collaborating within and among itself, among the projects and with other projects. So here we generated fish curatorial manual. We established a manual for curatorial techniques. And then we are going to publish an otolith atlas of marine fishes from Western Visayas. This is still ongoing, but it is progressing well. And then we have other outputs that are not lodged in any of the individual projects. I listed here 4. And then we also support the researchers and faculty members as they try to pursue their careers. For example, we're going to send one to Taiwan, one is going to Kyushu University, one is anticipating acceptance of his PhD degree in Tokushima University. And the last one, CIB grantee, just wanted to stay with us because he's looking at so many potential publications from the collections that we have. So a while ago, I mentioned that if we address management of fisheries, we're addressing SDGs 2, 1, and 8. But in reality, if we try to look at this is the reason why we are doing more in-depth studies of fish, more collaboration, why we need to support students, why we need to do partnerships, and this has to be done cyclically, continuously, so that we can sustain the growth of this particular sector, so that it can also contribute to the Philippine economy. These are our references. And before I close, I'd like to thank OVPA's EIDR team, the members of the EEMC. I'd like also to acknowledge the support given by our OVCRE's Leverage Research Fund. I'd like to congratulate and maybe thank members of my team, Dr. Malay, Dr. Luisma from the UPD MSI, Dr. Lumaino, Professor Gahe, our project manager, and to all of you who are listening here, thank you very much.
Oh, wow. Great presentation and congratulations, Dr. Ricardo Babaran, and to your entire team. No? You have so much accomplishments. Despite the pandemic, I know you have had so many problems. But despite that, you have collected the samples that you needed. You have characterized them. You have set up your museum, no? Magkaka-museum na sa UPV. This is really science working for even our fish, no? I'm so happy with that. So, thank you very much for that great presentation po. Do we have questions? Meron tayong congratulations po ulit kay VPAA, Cynthia Bautista, to your team. Ano ba tawag ni ma'am dito? We will rope or
rare rope. Hello.
Your accomplishments will become more meaningful for the Filipino people. So, questions? Meron po ba? Dr. Babaran, if I may, so itong pong lahat ng ito is to actually put emphasis more on ano ba yung mga fish natin na talagang competitive o may competitive advantage, and now we are actually characterizing them. So para ano yung mga fish culture ba yon? Sorry po, hindi ako masyado familiar, but how do we actually use all of this information?
There are three sectors of fisheries that are related to production. Sometimes they're treated like they are independent. If you look at aquaculture, we're only culturing very few species. And one of the reasons why we are not culturing enough fish is that we don't know much about their biology. And so Project 4, for example, we need to really study what their growth, reproductive capabilities are, so that we can bring them to culture systems onshore and support aquaculture. And aquaculture is important because this is a base for economic activity, economic development in various parts of the country. Remember, we are mostly water, and then we can create a livelihood out of this particular activity. But then the current policy of the government is to generate food to feed the growing population. That is okay, but the equal focus, maybe a substantial increase should also be made for pelagic fish, for marine fish activities, marine fisheries or capture fisheries.
Thank you very much po.
And we need to move fast because many of our species are already declining. We need to move fast, really.
Ano yun, nagiging extinct ba po yun? Ganun po ba yung ibig sabihin nun?
Yes po. No, we can promote the collapse if we harvest them without any regulation.
Oh, so you have to work with regulation also. I saw that problem. And
Yes.
so we hope we can actually address that. Ma'am May, do you have questions?
Yeah, Dr. Babaran, good afternoon po.
Good afternoon po.
Ito po yung program na hindi po talaga ito mapigilan na gumawa ng napakaraming projects. Ano po? And we're wondering, no? So kasi 2 years lang yung ano natin no? So and I think you will need a lot more time to be able to finish the objectives, no? To at least yung objectives for the 2 years.
Yes,
and because of the lockdown, talagang nahirapan kayo, no?
Yes, po. Especially Project 3 because we need to collect our samples from all over the country. And partially because of that, I got Omicron. So it's difficult. But we just have to move on because there seems to be no adjustment for EIDR's policy. So just kidding.
So hopefully be able to address as much as possible yung lahat ng objectives
Yes,
ng project ng program.
We'll try our best.
Yun po bang you talked about some of the species being aquacultured kasi hindi naman konti pa lang na aquaculture natin no. Doon sa pinag-aaralan nyo, may candidate na po pwedeng
the Cellar is one. The captors is another, but SIFDEC went ahead of us because I was not able to gather more of our UP scientists with our group. So, but there are other species that are good candidates.
I hope that
we need to study biology and feeding habits.
Yeah, so there should be also some kind of consultation between SIFDEC and the UPV, no? Kasi although SIFDEC is Southeast Asian, that's a Philippine government's funded agency, no? So para maganda yung pinag-aaralan nyo, hindi mag-overlap o at nagkakatulungan, kumbaga.
We are doing that po. But I think many of our scientists within UPB are also capable of doing the same things. We just need to support them, I think.
Okay.
Okay.
Marami pong salamat, Dr.
Thank you
very much.
Babaran,
Thank you
Dr.
very
May.
much.
We have Dr. Ano na nga?
Chef Maala.
Paala. Kayo na po.
Regarding yung project 3 News, sir, Dr. Babaran,
Yes, pa.
how much yung more time to accomplish this one? Kasi this is a very complex and complicated component of your study, the use of otoliths for age determination. You need a specialized or well-equipped laboratory for this.
Yes, po. The laboratory, we already arranged the analysis of our otolith samples from with Arizona State University. There is a lab there that processes chemistry of otoliths, and there's another lab that processes isotopes of in otoliths. There's a lab somewhere in University of Texas. So we already coordinated them, and we just have to send our samples for analysis to them.
So this part of your study cannot be accomplished here in the Philippines?
No, no. We don't have equipment that can do that right now.
Kasi if you're going to use otoliths for age determination, dapat may stock kayo na fish doon sa laboratory with known age. So from spawning to say hanggang 5 years or 6 years, kailan may reference samples kayo doon?
Sorry po, you mentioned the Project 3. I
think that is otoliths, di ba? Otoliths?
Yes, pa. Project 3 and Project 4 are using the same material, but Project 4 is more about age. Yes, I agree that there has to be a reference otolith lab to reference the age of fish. That's true. And we're moving towards that direction po.
So for example, if you are aiming to use tuna as your mga experimental animals, so ang lalaki yung tuna, so how could you house those reference fish in your laboratory?
We're not ambitioning to use tuna because it's very expensive. We're only targeting the other important small pelagic fish, for example, for this purpose. Kasi first, we don't have the capability yet to culture tuna in the Philippines. It's very difficult and it takes a long time. So not tuna ang target po natin, but we're targeting the other important pelagic fish species.
Kasi sir, doon sa Project 1 ninyo, examples nyo doon mga tuna, di ba mga identifications nyo?
Hindi po.
Then dito sa otoliths, hindi na kayo gagamit ng tuna, so you will be using other species.
No, sa Project 1 po, hindi tuna po yung example po natin doon.
Ano yung malalaking fish na pinakita mo doon?
No, Project 1 is more diversity, so maraming species ang involved. Project 3 is about connectivity of what our tuna resources with the tuna resources in the West and Central Pacific Ocean. So we want to establish connectivity so that we can probably leverage what we have to get more access to the tuna resources in the West and Central Pacific Ocean. So ganoon po yung connection. Project 4 naman ay gusto natin pag-aralan yung mas maraming isda dito sa Pilipinas so that we can support aquaculture and address yung Agenda 1, SDG 1, SDG 2, SDG 8.
Okay, thank you sir. Thank you sir.
Thank you po.
Thank you po, Professor Maala, and thank you, Dr. Babaran. There's really a lot of good research work going around dito sa ating UP system, no? Thank you very much for all the support, and thank you for our scientists who work very hard despite all the challenges especially with the pandemic. No? So let me end this session by thanking our 4 great speakers. We have Dr. Cecil Bayaga, we have Dr. Lilibeth Reyes, Dr. Cheryl Ariola, and of course Dr. Ricardo Babarana. Thank you all for your support, for all your wonderful comments. So thank you very much.
Thank you very much, Dr. Nina, for leading us on our first set of presentations this afternoon. Congratulations to Dr. Cecil, Dr. Lilibeth, Dr. Cheryl, and Dr. Rick, and your research teams for your accomplishments. We are now through with the first session, but before we proceed to our session 2, we will take a 10-minute break. We still have 2 presentations in our next session, and our chair will be Dean Joy Abrenica. See you after 10 minutes. We hope that you have taken your bio, coffee, or water break. Let's resume for the second round of presentation for today. Let's welcome our chair for this session, a member of the EMC and the current Dean of the School of Economics, UP Diliman, Dean Maria Joy V. Abrenica.
Thank you, Fatima. Okay, they say that we always save the best for last. So there we have last two papers, but I hope you will still pay attention and find the research findings interesting. So the first paper is entitled The Development of In Vivo and In Vitro Models for the Study of Endometriosis in the Philippines. This will be presented to us by Dr. Michael Velarde, professor from the College of Science, UP Diliman. Dr. Velarde, the floor is yours.
Thank you very much for this opportunity to be able to talk about our EIDR program projects. Our program is focused on the in vivo and in vitro models to study endometriosis in the Philippines. With this, we have two projects, one from UP Diliman, Institute of Biology, and the other one is in UP Manila at the Department of Physical Sciences and Mathematics. In UP Diliman, we are establishing a mouse model to mimic what's occurring in vivo in humans in terms of endometriosis. And so the idea is that if we develop this mouse model, we are able to develop and understand the etiology of the disease. In UP Manila, they're looking at the different food groups, different particular vegetables, a different part of the Filipino diet, and looking for phytoestrogens in that diet that might be helpful in terms of inhibiting the progression of this disease. So the team is composed of Project One staff, with me as project leader, and Dr. Joyce Ibana as one of our project staff helping us with the model. And we have research assistants and also lab aides working with us. And also for Project Two, we have Dr. Nicolas and Dr. Kiming, who's working with us, and also the research assistant. This is also in collaboration with University of Arkansas for Medical Sciences, Dr. Rizal, and helping us with our project on endometriosis and also the mouse model. We have mentored several students, PhD students, and also MS students. Some have already graduated and already moved on, and some are still continuing with us. And some even have created a project, spin-off coming from this project as well. So what is endometriosis? I'm not sure if everyone knows what endometriosis is, but endometriosis is a disease that that can present as an ovarian cyst. So sometimes you may hear it as an ovarian cyst or chocolate cyst forming in the ovaries. And this cyst, this endometrioid, can sometimes metastasize or move and grow at other parts of the peritoneal cavity, something similar to cancer. They're not necessarily considered as cancer, although these endometrioid can increase the risk of development of cancer. They can grow in all the different parts of the peritoneal cavity. And 1 in 10 women are estimated to be affected by this particular disease. It is very difficult to diagnose because to confirm the disease, you have to do surgery in order to see if it's indeed endometriosis. However, there are some clinical presentations that could indicate that maybe you have endometriosis. The problem with endometriosis is cause a lot of pain for women. Imagine endometrial tissues growing elsewhere in the body, and during menstruation, it will bleed out because of the cycle. So that can cause a lot of pain for majority of women who have this. In addition to that, this can also result in problems with pregnancy, problems with potential growth of ectopic pregnancy, and can cause miscarriage, preterm birth, sometimes even associated with developmental problems in the child, such as small development of the child. There is no cure for endometriosis at the moment. There is a treatment, surgical treatment that can be done, and there are some over-the-counter medications like oral contraceptives that can be used, but this is just to manage the disease and not necessarily cure them. So hence, we are interested in looking for alternatives to this, especially for those women who are affected by this particular disease. So the idea of the project is that for project 1, we will develop a mouse model that will mimic what's happening in humans, that we will grow endometriosis in the mouse. And then we also, for project 2, we will then look at screen the different types of diet from vegetables, from seeds, from different plant products that can be obtained, and then look at their composition and see if there might be a potential compound there that could help reduce the gravity of the disease. Or maybe sometimes, hopefully not, no, there could also be some chemicals there that might promote or aggravate the disease. So it will be important to see which diets we would avoid, or which diets should women take to prevent this disease from occurring. So that's basically the idea of this. It is a preclinical trial model. We will just look at using mouse and in vitro data. So this is just a iteration of the specific aims of the project for 1 and 2. So for project 1, we were able to develop different mouse strains to induce endometriosis. We tried different strains such as ICR mouse and also the C57 Black 6 mouse. We even tried a transgenic mouse to see their development of endometriosis. And you can see that with this mouse model, we are able to reproduce very similar structures that can be found in women with endometriosis, showing these classic gland-like structures that is reminiscent of an endometrium, but they're not in the endometrium. They are in fact growing in the peritoneal cavity. It depends on it depends on the strain of mouse. It's just interesting that some strain of mouse will develop endometriosis in subcutaneous, and some would prefer anywhere in the peritoneal cavity. So that tells us that there are maybe genetic differences, contribution of genetic differences on the development of endometriosis. And indeed, what we saw is that differences between the mouse strains, the ICR develop lesser endometriosis compared to the black mice. We've also seen some of the genes, the difference between the two. The ICR mice seem to have more inflammation and more immune response early on. So maybe the immune system is preventing the development of this endometriosis, suggesting that the immune system plays a major role in preventing the development of endometriosis. In Black 6 mice, they tend to be lower. What is also interesting is that in the strain of mice, in the ICR mice, they don't proliferate as much compared to the Black 6 mice, suggesting that there is also an intrinsic ability of these endometrium. If you have an endometrium that grows so much or proliferates so much, then your chances of getting endometriosis also higher. We also started trying some of natural products, such as genistein, on the development of endometriosis. We're still on the increasing the number because initially we didn't really see any difference at the moment. So hence, we are screening for more compounds, and that could help in terms of inhibiting the mouse, although that might be beyond the scope of this study. So all of this, in terms of the development, the mouse is being prepared for a manuscript for submission. There are all other deliverables for project 1. I just don't have time to discuss them. But for project 2, there are what they're doing is that they're analyzing the quantification of these compounds, these phytoestrogens, such as genistein that I mentioned a while ago. Then looking at their levels of the amount of compounds found in our diet. So the different plant samples. So they're able to develop the model. This is just a sample of that, and they were able to quantify it from their plant products. In addition to that, they are able to develop a yeast-based assay to look at the ability of these compounds to promote estrogenicity, because estrogen promotes or aggravates the disease of endometriosis. So if you have a lot of estrogen, then that means that they may potentially be developing more endometriosis. On the other hand, if you inhibit estrogen, then you may be able to inhibit the development of the disease. However, interestingly, some of these compounds can have dual effect. No, they can have promoting the estrogenic activity, but the same compound at certain doses can also inhibit the effect of the that estrogenic activity. So they were able to use the a screen for that for the compounds using the yeast-based assay, and then in the help with our lab, we are also able to develop the a screening using a mammalian-based cell culture assay. Okay, so I'm just going to skip through this. We have identified several fractions and that might be that might have estrogenic activity showing here, and then we also have that I did not show here that have anti-estrogenic activity. So the significance of the study, we're able to develop an in vivo model as well as an in vivo screening platform so that we could have a platform for a drug screening and development for treatment of endometriosis, and even identify and screen for plant products that might be helpful in terms of our diet, for women who have endometriosis. The idea here is that moving forward, we are applying for grants. So in collaboration with PGH, we are developing preliminary data on clinical samples to study endometriosis in women and maybe correlate them in terms of their diet as well, and see if diet can have a major contribution to the development of the disease in humans. So in addition to the in vivo assays and in vitro assays and mentoring of students, we have been collaborating with Dr. Rosie Simen at UAMS. We're able to send a student there to give to do experiments and to learn some of the techniques that they have done in terms of the development of mouse models of endometriosis, as well as cell culture-based assays to study estrogen activity. And one of our students was able to present an international conference, and we also have presented in other local conferences as well. Currently, we have published 2 research articles. There are still 3 manuscripts ongoing for the deliverable of the project. So this is just a sample of our publication. The main SDG that we are addressing is related to good health and well-being, because we are focused on clinical research. So I would like to thank the lab, who this was taken before the pandemic, where we still have some team. Some of this been some of them are part of the EIDR project, and some have been, because we're fortunate that we have the EIDR, we were able to secure some other funds from outside as well. It created a seed grant for us, so we were able to acquire some funding from other sources like the USD. We also think for Cooperate to be able to send one of our students in UAMS to be trained. Of course, we think of our other project, Dr. Kiming and Dr. Nicolas. Thank you very much.
Thank you, Dr. Velarde. Okay. Oh, all right. Immediately, do you have a congratulatory note here from VP Cinch Bautista? She said that, well, it's indeed a good presentation, very lucid one, and hopefully it will help us develop, or it will help in the development of treatment of the disease. Okay, so are there questions on the floor? If yeah, I don't see any question at the moment, but may I ask, since diet seems to be very important in all of this, are you thinking of collaborating with your American university counterparts to study how different Asian diet is with that of Western diet that may in effect contribute to the disease? Yeah.
Yeah, actually, that's a very interesting idea. We have not initially started yet in terms of comparison between the US, for example, or doing the Western diet and the Filipino diet. The problem, one of the problem is the West Filipino diet apparently seems to be close to the West diet in the different parts of Asia. And so we are looking at the particular diets that we are taking in instead, like the certain vegetables that we have. Is that any different? So we could do that. Another thing that we were thinking about is maybe we can compare our diet with other parts of Asia instead, because the Filipino diet seems to be, as I said, very close to the Western diet. And that's probably also maybe the reason why that some anecdotal evidence that Filipino endometriosis seems to be worse compared to other Asian endometriosis. So maybe it is better to compare it with Asia instead of just simply comparing it with Americans.
Okay, thank you. That's good to hear. And of course, as a woman, I'm very interested in your finding. But since there's a certain age limit by which you seem to contract the disease, you said it should be childbearing age. So if you're way past that age, you're a little bit safe. Is that could we infer? Could we make that inference?
Yeah, typically, once you pass a certain age, then the disease is not as high, the risk factor. Although the problem is if that now turned into cancer, that would be a problem. Okay. So hopefully it didn't turn into cancer.
All right. Okay. PMA. Sorry. Do I have another question here? Okay. Okay. Yeah. Yeah. Oh, okay. PE Maala.
Okay, Dr. Marla.
Yeah.
So, are you going to make a recommendation, sir, that those plants with high estrogen content be banned or prohibited from being eaten by Filipinos?
Yeah, I don't think I can already say to ban certain type of food to be eaten because this is one particular disease, right? So it's more of a recommendation. But at the moment, we cannot do that without a clinical data. So hence, that's why once we have the screening, we can then now justify of what particular survey, maybe questionnaires that we can ask to pinpoint if they are eating this type of diet and do they have correlation with that particular disease, with the endometriosis. But we have to collaborate with PGH and have that comparison between the two before we can recommend, because these are just in vivo and in vitro studies.
I observed that many of these studies one of the outputs should be a policy statement, di ba? So ang daming mga studies na like yours. So kung if you will find out that this type of vegetable is risky in terms of resulting to endometriosis, then there should be policy on the part of the government to be careful about eating these types of vegetables. Yun lang yung point ko, sir. Kasi what is the use of establishing that this type of vegetable or food is has a high hormone mimicking substance resulting to endometriosis kung wala naman action ng government. So I think a policy on this statement is an important output of such a study.
Okay. So we'll take note of that. I think that we usually don't say policy when it comes to medical. It's more of a statement, a statement and a recommendation of what to eat and what not to eat.
Gaya ng dietary intake, di ba, kanina?
Yes.
So there should be a policy statement, one of the outputs of such a study, that to have a healthy baby, ito ang dapat kainin ng mothers or what? Kasi otherwise, what is the use of such a study kung wala namang mag-implement ng mga results ng study na ito? And the one way is to come up with a policy statement, something like that, by the government.
Yeah, but I think, Dr. Velarde, your team is looking more at finding more treatment. Is that correct? I mean, better treatment, or because you said there's no cure, but it's only treatable but not curable. So, so maybe the intent, and I see that this is just preclinical, and therefore long ways to go before you could actually come up with some recommendation for better treatment of the disease. Is that correct? Is that correct?
That's correct.
Okay.
That's correct. Yes. Although I do want, I'm very also much interested in policy. I think it's very, very important that if we develop a policy, but in order for us to go there, we still need to have the clinical side, and that is the next phase of this. We called it a phase 2, is to work with clinicians on developing that comparison, especially if we have clinical data, and that will be stronger to say, oh, we see now there's a strong correlation, so we need to have a good recommendation and a policy statement, perhaps.
Okay. Thank you. Thank you, Dr. Velarde and your team. And if there's no more question, maybe, okay, I hope, well, we look forward to the results of your succeeding phases of your study, and we hope you could do more collaboration with your partners in the foreign universities. Thank you, Dr. Velarde. Okay. So we now move on. Okay, we now move on to the last paper, and this indeed is multidisciplinary. Okay, I see some names in the project team who I'm familiar with. So, okay, the title of the project is "How Safe Are Our Roads? The Components of Post-Crash Response, Safe Roads, and Safe Vehicles in Road Management." Okay, and it's quite a long title. So, Dr. Chinky Lu of the National Institute of Health from UP Manila will do the presentation. Dr. Lu.
Good afternoon. I am Jinky Leilani Lu from the National Institutes of Health, University of the Philippines, Manila, and I am the program leader. We are glad to present our report for an important and timely research program sponsored by the EIDR OVPAA UP system entitled "How Safe are our roads, the components of post-crash response, safe roads and safe vehicles in road management, road safety index, and road legislation in making our roads safer. It has 4 component projects focusing on 4 disciplines: public health and medicine, engineering, statistics and modeling, legislation, and management. All these disciplines are integrated into the program to come up with a multi- and transdisciplinary research program. The 4 component projects are: Project 1, the epidemiology of cases of road crashes and injuries at the Philippine General Hospital, led by Dr. Herbosa. Project 2 is investigating the role of safe roads and safe vehicles in road safety management in the Philippines, led by Dr. Ricardo Sigua. Project 3 is constructing an index for road safety using Metro Manila baseline investigation, led by Dr. Dennis Mapa. And Project 4 is investigating the current road management and legislations in the Philippines, led by Dr. Jinky Lu. According to the World Health Organization, road traffic injury or road crash is one of the leading causes of deaths around the world. The magnitude is as follows. There are about 1.25 million deaths globally due to road traffic crashes, which is an estimated 3,400 deaths every day. Half of these deaths are among vulnerable road users, which consist of the pedestrians and motorcyclists, those in the lower income group. With the need for a global guideline in reducing road crashes and promoting road safety, the United Nations General Assembly came about with a decade of action for road safety covering the years 2011 to 2020. The Decade of Action Plan has 5 pillars. Pillar 1, road safety management. Pillar 2, safer roads and mobility. Pillar 3, safer vehicles. Pillar 4, safer road users. And Pillar 5, the road post-crash response. Hence, the birth of our research program. We have several objectives for each of the component 4 projects, as you can see on the slide. Suffice to say that as a whole, this research program is aimed at investigating road safety in the Philippines using evidence-based research and guided by a framework that captures all aspects and components of road safety. The program is also aimed at developing a road safety index, or RSI, using data from Metro Manila, which is the most problematic relating to road crashes, road injuries, and fatalities. Altogether, this program is for making roads safer using the Safe Systems approach. Here are some highlights of the various research projects. For Project 1, the total number of cases of road crash patients brought to the Philippine General Hospital is 4,979 from January 2008 to December 2017. There is a 426% increase in road crash patients, and that is an average increase of 21% annually. The mean age of road crash patients is 30.8. However, the age distribution is bimodal, as you can see in the figure, initially peaking for 6 to 7-year-olds and the highest peak at 24-year-olds. These are the very young and the working population. Majority of road crash patients in PGH are within the 20 to 29 age range, followed by those who are in the 30 to 39 age range. Again, these are the people within their prime working age. 61% of the reported victim types are drivers, 28% are pedestrians, while 11% are passengers. 77% of road crash patients is due to motorcycle crash. Alcohol intake is the most reported risk factor, followed by non-use of helmet and non-use of seat belts. However, it must be noted that there are several missing data on this variable. A huge majority of patients brought to PGH are discharged with an improved or recovered status. 378 are discharged with unchanged in status, while 166 died. 85% of patients brought to PGH suffered injuries in multiple body parts. Most commonly reported is external injury at 45.9%, followed by injury to the extremities at 32%. Most commonly, patients have injuries in one or two body parts. However, 15.7% also have three or more than three body parts injured. For Project 2, here are some of the highlights. Information such as the annual average daily traffic, number of lanes, types of intersection are necessary in understanding the most dangerous roads in the country. Saan ang mga daan na nakakamatay? 3 corridors were studied, namely EDSA, C5, and Quirino Highway. These intersections, mid-blocks, and U-turn slots for each corridor were identified as black spots based on the MARAS database. EDSA has a daily traffic of around 364,000 vehicles. Meanwhile, C5 ranges from 178,000 to 218,000 vehicles per year. For the top 3 identified black spots at EDSA intersections, we have the following data. The location of Ayala Avenue refers to the 4-way intersection of EDSA, Ayala Avenue, and McKinley Road. The signalized intersection with 4 phases has the highest number of incidents of road crash with 2 fatal incidents among all the intersections along EDSA. For the top 3 identified black spots at C5 intersections, we have C5 Road, CP Garcia Avenue, which have the highest number of road crash incidents with a total of 46. The intersection of C5 and CP Garcia Avenue is a 4-way signalized intersection. Ortigas Avenue, a 4-way signalized intersection, has a total of 41 incidents. Kalayaan Avenue and Lanuza Avenue both have 38 road crash incidents. For the top 3 identified black spots at Quirino Highway intersections, we have the Bankers Village 2 along Quirino Highway, which has 13 non-fatal and 3 fatal road crash incidents. Sarmiento Street, a signalized 4-way intersection, records a total of 15 road crash incidents. Zabarte Road and Tandang Sora intersections both have a total of 14 road crash incidents. For the top 3 identified black spots at EDSA mid-blocks, we have Balintawak, which has 22 road crash incidents. The road along Balintawak consists of 6 lanes for each direction. Camp Aguinaldo on the northbound side along EDSA has a total of 19 road crash incidents. The road changes from 6 to 7 lanes approaching the flyover. For the top 3 identified black spots at C5 mid-blocks, we have along C5, the road along Bagong Ilog has recorded the highest number at 59 fatal incidents and 3 deaths. This slide shows us the following. First, the top 3 identified black spots at Quirino Highway mid-blocks. Second, the top 3 identified black spots at EDSA U-turn slots. Third, the top 3 identified black spots at C5 U-turn slots. Project 3 utilizes several databases on road crash data. So far, 1 database has been analyzed, and this is the Metro Manila Accident Reporting and Analysis System, or the MARAS, a road crash reporting system created and operated by the MMDA with the Philippine National Police. The total number of cases is about 1.35 million, which is actually a huge number of road crash incidents in Metro Manila alone from January 2005 to December 2020. Imagine having 1.35 million road crashes for the span of years. Between the years 2005 and 2019, the number of road crashes increased by 32.9%. On the average, This is an annual increase of 4.7% in road crashes. The lockdown during 2020 could have influenced the sharp drop in the number of total cases in 2020. Total drivers killed so far is about 2,700. Total passengers killed is about 800. Total pedestrian killed is about 2,600, and that is a total of 6,283. Per fatal crash incident, there is a total of 6,019 fatal crashes. But please take note that there may be more than one killed person per fatal crash incident. The final output of Project 3 will be the development of a road safety index for the Philippines. It is non-existent at present. The RSI will be based on the 5 pillars of road safety of the World Health Organization and the United Nations Global Action, considering the 5 pillars of action that we already mentioned. Altogether, the components, indices, variables, and factors to be considered for this road safety index are shown in this figure. Suffice to say that these are exhaustive and comprehensive in order to have a reliable road safety index. For Project 4, which is the final project of this research program on road safety, the highlights are the following. There are several laws existing already in the country in ensuring road safety and road user protection. The laws vary from speed limit, use of seat belts, distracted driving, drunk and drunk driving, motorcycle safety, children's safety, driver education and training, use and installation of accessory devices, safety of vulnerable road users, creation of agencies for road safety, and highway administration. Second, different regions in the Philippines consider promotion of road safety by establishing city ordinances, which often revolve around the strict enforcement of traffic rules and regulations, non-obstruction of public utility vehicles when using the road, and road users who utilize sidewalks and streets as vendor-friendly areas. Third, the current road management in the Philippines is continuously evolving and developing. Our initial data show the following significant outputs and impacts. Victims are the very young. Victims are people in their prime working age. Motorcycles comprise the vehicle type most involved in road crashes. Pedestrians are among the most vulnerable road users. Alcohol increases risk of death and injury, and non-use of a helmet and seat belts also increases death and injury. Increased fatality and disability in the 20 to 39 age group has economic consequences due to decreased productivity in this population. There is also a need to create a policy which will increase safety for motorcycle riders and their potential victims. There are two major patent or copyright potentials arising from this research program, and these are, first, the proposed trauma scoring system for a proper prognosis of trauma patients, particularly road crash or traffic injury patients brought to the hospitals. And second, the development or construction of road safety index in the Philippines. This is the innovation that will be generated from the program on road safety with 4 component projects: a new method, a new classifier, a new scoring system for trauma patients that will benefit hospitals and patients alike, as well as the construction of a new road safety index that captures the comprehensive and multidisciplinary aspect of road safety that will benefit our national agencies. There are no major issues related to the conduct of research activities except for the delay and concerns arising from the COVID-19 pandemic. For the SDGs addressed, we have the following. SDG 3, good health and well-being. Studies for road safety is a way to continue to improve health of the public, given that road fatalities and road injuries are becoming serious threats to general health and well-being of the Filipino people. We also have SDG 11, which is making cities and human settlements inclusive, safe, resilient, and sustainable. Target 11.2 ensures access to safe, affordable, accessible, and sustainable transport systems for all, with special attention to the needs of those in the vulnerable group, such as the women, the children, persons with disabilities, and the older persons. I would like to end this initial data presentation of our IDR research program by saying that road crashes do not happen. They are caused.
Thank you, Dr. Lu, for a very lucid and relatable this presentation. Okay, being not in the sciences, so this one I could understand very well. Thank you for that. So, is there? Oh, okay. There are already some messages in the chat board. Okay, if I could read. Okay, from BP Cinch, much appreciated. Jinky, calling you by the first name, and Ted and the team for providing, evidence for what would commonly assume, okay, for the program's policy implications and method and measures and indices. Okay, all right. Questions on the floor? If none, I'm very interested to ask. So I suppose you are correlating the increase in crashes, the sharp increase in crashes with the proliferation of motorcycles. But how does that compare with the crashes in other Asian countries where you have more motorcycles than what we have in the Philippines?
Dr. Ted Herbosa, can you answer that because it relates to Project One?
Yeah, that's Project One. And true enough, yes, the burden of two-wheeled vehicles on the road intermingling with other four-wheeled vehicles like trucks, cars, SUVs are the one that is the root cause of all of these motorcycle injuries, the 77% admission rate of our trauma cases at the Philippine General Hospital. We need to correct this. There has been some leeway that happened during the time of COVID. You will now see motorcycle lanes that have been implemented together with bicycle lanes that have created some level of safety to our two-wheeled riders. However, they're again being infringed upon with the alert level 1 and the heavy traffic and volume. We need to probably use policies as implemented when I worked in Malaysia. There is really a road that is separate for the two-wheeled vehicles. It's still on the highway, but there's a tree or an island between the two. So I think in the country, we saw this in Iloilo. Iloilo implemented a main road wherein the bikers were separated from the main road. So I think these are the implications of the study. We find the same things that are found in middle-income countries like us, wherein there is more use of the motorcycle because that's affordable, especially for young men who are still earning money. And the sad part is these young men get injured. The cost of their injury is tremendous. They are the ones that they cannot work. So we call it daily, the disability adjusted life years. So this is really a very important health and economic burden for the country. So very important yung findings nitong EIDR study na to.
Yeah. Any question on the floor?
Baka si Rick would like to add about the two-wheeled vehicles. He does studies with international agencies as well.
Yes, good afternoon. If I may add, well, here in the Philippines, the surge of the number of motorcycles started in mid 2000, somewhere in 2004, 2005, due to the influx of cheaper motorcycles. Of course, from where, but before that, motorcycles were very expensive, largely coming from Japan. So we came late. Our ASEAN neighbors have been using motorcycles ever since, and yeah, I think we have to learn from the experience of this of our ASEAN neighbors, particular Vietnam, Thailand, even Malaysia, where we have really millions of motorcycles. So we were somehow caught flat-footed in terms of policy because unlike before, most motorcycles were used as tricycles as a form of livelihood. Although now there's the challenge of this because motorcycles now are being used for Grab or for delivery. So I think the government has really put policy on this, although because of the pandemic, as Dr. Herbosa has mentioned, we have been providing motorcycle lanes. But another issue is with the speeding, and in particular, lane splitting of motorcycles, meaning riding at a fast speed in between lanes or in between lines of vehicles.
Okay. Well, the irony is that the growth of motorcycles is actually partly brought about by the motorcycle development program of the DTI. Is that so which is really part of industrialization program we have. So that gave rise to more motorcycles and on the road. But you also talked about road safety index, and I was wondering if you could use that road safety index, for example, to see how the roads inside the campus of UP Diliman in particular is safe enough so that we're advocating this carless academic oval. So is that something that is urgently needed at this time, given road safety concerns?
Yes, the project leader involved in the road safety index construction is Dr. Mapa actually, but he will get all the other data coming from projects 1, 2, as well as project 4. And this road safety index is really focused on creating one hopefully for Metro Manila, because the IDR is really focused on Metro Manila situation or scenario or milieu. And of course, we can use the RSI, the Road Safety Index, for specific locations like UP Diliman campus and any other road areas in Metro Manila.
Okay.
May I add that the road safety index was envisioned as a measure. So you can quantify any intervention. So for example, you're going to implement a carless in the academic oval, then we will use the road safety index before and after the intervention. So you'll have something numerical or objective that the intervention you did really saved lives and prevented injuries.
Okay, that's good to hear. So that's one way of measuring the cost and benefit of making the academic oval carless, which is of great interest to us. Okay, okay. One more question from Dr. Lily, Professor Lily Tope. Yes, please. Okay, would you like to open your mic, or should I just read your question? Okay, does pedestrian activities figure in your research? I think yes, but anyway, please, especially those who do not know how to maybe cross the road or observe all this road rules. Yeah. So yes.
Yes, pedestrian as a variable in project 1 was measured, no, in terms of the number of deaths or the number of cases first, and then the number of pedestrians that eventually died due to the road traffic injury or the road crash. And there are other variables that we considered, and of course the type of road user, whether pedestrian, passenger, or driver, is being considered in our data analysis. And one of the major outputs of our program is eventually to come up with an IEC campaign material to also include the pedestrians. And it's very important that we emphasize the need, no, for pedestrian lanes, and that should be respected also by other road users.
So I agree, no? Technically, all of these are going to change with the behavior, no? That's another part of our study actually. So we created some videos actually also with this project to try to inculcate change in behavior. I think that's the important part, and pedestrians and drivers are the target of these measures, not only IEC, but really making them change behavior. Yung making them really cross in areas that you need to cross, and not take shortcuts and put risky activities, because they end up at the Philippine General Hospital when they do this.
And we are trying not only to change the behavior, no, on the roads, but also to make road safety a culture among the Filipino people, because we are all road users. When we go to the grocery, we go to church, we go to school, to our work, no. So this is a very important research program.
I am told to wrap
You're going to
up.
go to elections. You ask your local chief executive who's the candidate what their road safety platform is.
Oh yeah, yeah. We better do that.
Yes.
Yes. Thank you.
Yeah.
And I suppose in the next Congress we'll see a Magna Carta for road safety coming from your group.
Yes.
Okay.
Sigwa.
Okay, that would be
Yes.
good. That would
Yeah,
be a good one. Yeah. That would
Yes,
be a-
our pedestrians are still our most vulnerable road users. So
Yeah, okay. I'm sorry to cut this discussion, but I suppose we will have some more questions for your team later on. So Fatima, I give the floor back to you.
Thank you, Dean Joy, for moderating our session 2. Congratulations to Dr. Michael, Dr. Jinky, and your research teams for your accomplishments. Just like yesterday, where we had a meaningful discussion on our presentations, we are now in our open forum to discuss the research topics presented to you this afternoon. To moderate the session, please help me welcome Professor Emeritus Seferino Maala.
Good afternoon, everyone. I am Dr. Seferino P. Mala, who will be moderating this afternoon's open forum. Congratulations to the presenters. Very interesting topics and results. And siguro hindi lang ako yung magsasabi nito. We're having information overload. 6 very interesting topics to digest. Now for the open forum, we have 25 minutes, and this will be free for all, meaning that, for example, if a participant wants to ask question from number 1 speaker, or number 4, or number 6, wala nang particular order. And I hope that the previous presenters are still with us to answer questions from participants. And those shy participants can key in their questions chat box. And now, who would like to ask the first question? Anybody? If none, then ruling dito, the moderator would be the first to ask question. And I would like to ask you, this question dun sa last presenter, what time of the day usually ba nagkakaroon ng mataas na incidence of road accidents? Anybody from the presenters or members of the staff? I don't know, but hindi ko narinig yata na mayroong data mention on that. What time of the day ba na usually these road accidents happen?
So,
Yes, that's also in our database, and in our database, most of them are happening in the early evening, or even late evening. We have many of these motorcycle injuries being treated at the Philippine General Hospital. Although we also have cases in the mornings during rush hour, but most of the cases really are at night when we get these injuries. Rick, what are your other findings?
Sir. If I may, it really depends on the severity of the incident. During daytime, when there are a lot of vehicles and the traffic volume is quite heavy, we may have a lot of property damage incidents, meaning involving vehicles colliding with each other. But the more serious ones would happen at night, especially late at night or early in the morning, when there are a lot of factors. One is visibility, and the other could be, the practice of drink and driving. So, and in those cases, most incidents would be severe, meaning fatal or having major severity.
And sir, usually yung nababasa natin ay this involves celebrities, di ba?
Yes.
Yung nagkakaroon ng accident sa gabi or early morning, and usually due to nasa sila ng alcohol,
yes,
or
sir.
Lasing or what?
Yeah, we know that from news, but in the database, we can. These are not really reflected. But we know from the news clippings that there are a lot of celebrities.
Yes, ma'am. Yes,
please. I also add something, that is in regard to time, but we also have data in regard the month, the month of the year, where we have the peak of road crashes. Based on our data, contrary to other reports where they say that road crashes are in August, which is the school opening, December, which is the holiday season, and summer season as well, our MARAS database shows that road crashes occur consistently across the months of the year.
Yes. Interesting. And another one, follow-up, ma'am, do you think itong pandemic will reduce the incidence of road accidents?
In fact, our data already shows that there is a dip in the incidence of road crash in 2020 because there are lesser vehicles on the road. So probably we can say that that is one of the good things brought about by this pandemic. But of course, we still need to steer up our economy and go back to the norm. But we really have to focus on the road management so that despite the fact that we are back to our normal activities, then we can still prevent road crashes and reduce the incidence of road crash.
Thank you for that information, ma'am. I can see academician Mendoza raising your hand.
Thank you
Please,
very- thank-
Ma'am Mendoza.
Yeah, thank you very much, Prof. PESF. This question is for our road experts. No, tinanggal ko yung mga accidents. But anyway, no, hindi ko ata nadinig, but ang concern ko yung many of our roads, whether major roads or minor roads, walang daan para talaga sa mga tao. Yung sa right or left side, no, yung hindi yung crossing. And I live in Los Baños. So talagang kahit bago yung road na ginawa, sabi ko, bakit po walang provision para sa pedestrian samantalang ang dami-dami na nagdadaan dito sa road na to, naglalakad lang sila. Pwede bang ilagay din natin sa report yon? Kasi sa mga experience ko sa Japan, kahit na maliit na road, meron siyang maliit na path na merong talaga sign na one person sa kanya yon. No, sana masama nyo din po sa report nyo. Kasi I'm sure na maraming aksidente ding nangyayari doon sa mga pedestrian natin na naglalakad lang. Thank you po. Congratulations sa trabaho.
Yes, yes, ma'am. Yeah, that's commonly observed, ano, yung pagkukulang sa mga provisions ng facilities for pedestrians, but wala tayong participants from DPWH, ano, but with our national transport policy po ay talagang binibigyan ng importance ang active transport. Meaning, ang nagbibisikleta at ang mga naglalakad. Ano? And nandun po sa polisiya na lahat ng dapat na bago ng mga highways ay magkakaroon ng provision para sa sidewalk at sa mga sikleta.
I'll also add, Rick, no, I was just in a workshop the other day. It's called Tactical Urbanism, sponsored by World Bank. And in Tactical Urbanism, they are redesigning. For example, that particular workshop was redesigning Caruncho Avenue of Pasig, so that the road and the existing road can be used by the pedestrian, by the cyclist, by the motorcyclist, by the cars, in a way na it will continue to improve mobility as well. So ang always complaint is, if you give more space to the pedestrians, nagagalit yung mga cars and jeepneys and public transport. But actually that study proves it doesn't. And in other countries, when you give more space to the cyclists and to the pedestrians, you actually improve the mobility kasi less people will use cars. They will just walk and cycle. So I think that's a good idea to also put forth in our findings and add that to the suggestions in road engineering design. Thank you,
Okay,
ma'am.
thank you for your comments. Mayroong suggestion dito that we should entertain also questions para sa mga ibang speakers or topics or subject matters. So sorry for this, but siguro mayroon ding interested doon sa mga previous papers or presentations. So Dr. Mendoza, is this for previous topic?
Yeah, follow up. Ano yung I think it was mentioned by VP since yesterday na isang achievement ng program, EIDR program, yung pag-upgrade ng mga laboratories sa buong UP system, no? And I think nakita natin sa project ni Dr. Lilibet, yung mga gamit na mga equipment nila ay talagang world class, no? Doc Lilibet, can you tell us ano ba yung bag sa ano na yon, sa IC na lahat yun available? Please tell us.
Yes, yes, ma'am. So we have for example, the high resolution mass spectrometry, which we use for the global molecular networking, is now in IC, and this is a shared facility. And actually, we also, our group also conducted a workshop on doing the analysis for molecular networking. So not just only the infrastructure, but even yung our group, yung training, we also shared that. I think we had about 200 participants from different institutions who participated. That was in March of 2021 last year.
That's great. I was really amazed yung yung mga molecules nakikita nyo doon sa it was nung palagi ko sinasabi, hindi pa ito naturo sa amin noong ako nag-PhD.
So and may I also share, when I was a grad student, kahit nung time ko din po, we didn't have these facilities. And so I'm very happy when we came back, all four of us are US trained scientists. So when we came back here, we have these facilities, and now we can do this also world-class research. So we're very thankful for that, for the support of course, UP and DOST as well.
Yes, of course. Yung kay Dr. Babaran din, Sir, ang ganda na ng facilities ng UP Visayas, and yung trabaho nyo, nakita natin na from barcoding, SNPs, SNPs, and then sa otoliths, no? So, and then of course, the morphological is very important. Hindi po pwedeng mawala yung morphological characterizations, ano po? So, ang ganda na rin ng facilities, sir, ng Dr. Babaran, ng ating UPV.
Actually, ma'am, we leveraged what we have already at UPV to get additional support from EIDR. Yeah, but some of the instruments we have ay walang patungan po. So kung pwede, yung savings ng year 1 namin ay ibigay na lang dun sa amin ulit para lagyan namin, patungan namin ng nabiling
equipment si Dr. Babaran.
Kaya if you remember, Ma'am Mendoza, one of my questions nung dun sa ni Dr. Babaran ay kung yung mga ibang projects niya were actually done dun sa laboratory nila. Kasi one of the objectives is to rehabilitate and update, di ba, yung existing laboratories nila doon. Because doon sa otoliths part ng study, that requires a very sophisticated
Pero
laboratory.
Maraming pong namin ginawa po doon sa labs namin. Pero the things that we cannot do, we really have to send the samples for analysis abroad.
Yes, I remember that for Dr. Babaran's big program, no. So for the otolith, yung talagang chemical analysis was really meant to be done outside. Wala pa, they're still just building up their capabilities. Pero yung morphological kaya, no. But yung sa chemistry po, so sa labas pa gagawin sa iba. And I think that's the way to go also. They're building their capabilities. So siguro later on, it's really necessary for us to build the otolith chemistry capability in DUPV. Siguro, but I think masyadong malaki ang gastos doon. But DOST will have the money for that probably.
We're always leveraging what we are doing. So for 20%, 80%, ganun lang po yung aming parating gamit na style. Yes.
And I think isa namang emphasis ng din ng EIDR yung to form collaborations, no?
Yes po.
To strengthen us. So pero dapat medyo equal din yung partnership, yung hindi lang tayo ang kumukuha. Dapat meron din silang makukuha from us, mas gusto ko yung gano'n na more or less equal ang partnership.
Yep. Yes.
So any other questions, Seth?
Kay Dr. Velarde, sir, mga participants, any question para kay Dr. Velarde? Kasi very interesting din itong topic na ito. Ito yung estrogenic plants or vegetables, So, Sir Velarde, are you coming up with a list of plants kaya or vegetables na may potential na causing this endometriosis. Para alam yung public what to eat and what not to eat. Sir Velarde, naka-mute ba kayo or what? Sir, can you hear me?
Baka wala si Dr. Velarde.
Nawala siya. So, parang nawala si Dr. Velarde, how about yung dietary intake ng mga mothers postpartum? Interesting din yung topic na yun, Any questions or comments?
Prof. Seth, Dr. Velarde is back, I think. Mike?
Ano na siya? Sir Velarde, still no response from him. Nawala na. So how about yung paper on yung dietary intake? Any comments on that presentation? Yung defect the bacterial communities and chemical compositions ba ng milk from 0 to 40 days postpartum? Anybody? Kung wala, we could go back again to the road accidents. We should trending as well as we do. Yeah. Again, question on dito sa road accidents. So, any other questions or what on the last paper? Okay, I have another question. May effect ba itong increase in gasoline price sa incidence of road accidents? Kasi baka dahil mahal na ng gasolina, so people will be driving less or going out. So what is your take on this or what?
If I may, sir.
Yes, please.
One of the major factor or exposure or sorry sir. One of the major causes din ng mga occurrence ng road crashes would be yung tinatawag nating exposure, no? Meaning nabanggit na nga ni Dr. Lu ito, yung traffic, no? And if you look at the siguro relationship din ng exposure at saka ng occurrence ng road crashes, the more na may traffic tayo ay the more ang nagkakaroon tayo ng road crashes. So it's not that linear, but some studies may pagka-exponential nga ito. So kung liliit o bababa yung level ng traffic, then expected na bumaba din ang ating bilang ng road crashes. But then, nabanggit ko nga na most likely ang mga bababa ay yung minor severity. But, kasi experience din natin na konti nga ang sasakyan pero mabibilis. So, pag nagkaroon ng incident ay malaki ang severity.
And I'll also add, maybe I'll
yes,
also
please.
add that, when the gas prices increase, some people start to think of using the motorcycle. And you saw in our study, 77% of our crashes are actually from motorcycle injury. So they come from a very safe car, they move to the two-wheeled vehicle, then you'll see. That's the correlation I would like to speculate. Right now, we've not done any study, but that's the speculation I would think. As prices increase, some people will not move in one car. They will just ride their motorcycle. So yun yung problema. Next.
So I think Dr. Willard is back, and also are there any questions or comments? Ma'am Mendoza?
Okay, thank you. But I think since Dr. Villard is here, why don't you ask him again the question, and
Yes,
Dr. Villard
okay.
can answer.
Nung wala ka, Dr. Velarde, I asked this one, are you going to come up with a list of plants or vegetables with their estrogenic components or potentials, so that the public will be informed what to eat and what to avoid?
We will come up with a list. Definitely, we will come up with a list. Kaya lang kasi, ang challenge nga, as I said before, is before we send it out to the public, we have to check first if that will be correlative sa tao. No? So if it's correlated doon sa clinic. Kasi sa amin, ang number one goal namin are the components of the food pa. No? So hinihimay kasi namin yung product eh. Parang the same siya kay Dr. Lilibeth when they are looking at the compounds and for the purpose of drug discovery. So yun yung kasi yung intent nito for treatment. Kaya hindi namin siya makuha as a whole, no, the full plant yet. It's just the main component. But that's the ultimate goal that we would like to do, is once we identify these components and these plants that may be good or bad, titingnan na namin siya at a bigger scale, no, at a gross scale, na if the food itself, in fact, iniisip na namin how do you prepare it, no? For example, when you cook it, is the compound still there, no? That's challenging. Baka mamaya pag niluto na, iba na. Yon. O kaya pinrocess ng differently, or ini-store mo siya, finreeze mo siya, it's different. So those are the things that we are also adding to the research para when we inform the public, it will be more informative. It's not just simply saying that talbos ng kamote is good for you. It's too simple.
Okay. Thank you for your answer, Dr. Velarde. So, any more questions or what? Yes, Ma'am Mendoza.
Thank you very much, PE Seth. Ang question ko siguro po pwedeng masagot ni Dr. Leia, Cecil Leia, as well as kay Dr. Cheryl, no? What will be the next steps, no, para for the application of the results of your research? Kailangan ba ng may verification? Tapos anong pupuntahan yung agency? Meron bang national agency or LGU ba para magamit, no? So yung kay Cheryl parang application na for is it commercialization kung po pwede doon. Kay Cecil naman adoption ng agencies, meron ba doon food sa nutrition, anong agency ang pupuntahan niyo para makakuha kayo ng support for the results of your research, para makita nila application naman ng research? Thank you.
Sige po. Mama, una na po akong sumagot. Yung nabanggit ko po kanina with Dr. Gloriani, yung tanong po niya, what's the next step to trickle down the results na po in terms of its applicability to the community, kasi sila naman po talaga yung magbe-benefit, the mothers who participated in our study. So we already have some initial plans on how to relay the scientific results to the community sa Makati po. Kasi sa Makati po kami nakapag-kolekta ng aming mga datos. So sila po yung very much willing and yung support po nila sa amin na ibigay in terms of the recruitment. So we plan to talk to them and then plan out or iron out a nutrition education program for the mothers in the barangay health centers. Kasi ang maganda po doon, kaya din po kami doon pumunta. Kada barangay po kasi sa kanila, meron pong health center. So it would be easier to plan, paano po ba, information dissemination. And also, one of the plans din po of the team is to give probably, I don't know, a summary, para pang po bang ganun, a summary of the results or the highlights to the National Nutrition Council, who is the coordinating body of all of the nutrition programs in the Philippines. So probably if they would have certain data on the results of the study, then probably they can use it, especially for the first 1,000 days of life, dahil ano na po ito batas na po to strengthen the first 1,000 days of life, na kung saan nakapaloob po doon yung lactation. Kasi the first 1,000 days involves the 9 months of pregnancy until the first 2 years of the child. So
I think Ma'am Mendoza,
yes.
Meron na akong notification dito from yung staff natin that I have to close this yung open forum kasi medyo na tayo. Overtime
tayo.
And we still have to listen to Ma'am Di Malanta for her closing remark, as well as yung certificate of attendance or what. Okay. Thank you. Thank you very much. Anyway, magkakakilala naman tayo. So if you have any more questions sa isang then pwedeng message or Facebook or text for your questions. So thank you very much, and very interesting open forum. Sayang bitin, kulang ng oras. So
Yes,
Ma'am, have the floor now.
Okay, and for Dr. Ariola, who hasn't responded to the question, you may post your answer to the chat box to respond to PMA's question. Thank you, PSF, project leaders, and the EEMC for an interactive discussion on our day 2 of the research symposium. Thank you also to our participants in Zoom and YouTube for your comments and questions. To present the certificates of appreciation for our speakers, may I call on Rona Abisado.
Thank you, Fatima. To cap off our day 2, the Office of the Vice President for Academic Affairs presents this certificate of appreciation to our speakers for imparting their invaluable knowledge during the 2022 Research Symposium held via Zoom, given the 7th day of April 2022, signed by VP Maria Cynthia Rose Banzon Bautista, to Dr. Cecil Lea T. Bayaga, to Dr. Lilibet Salvador Reyes, to Dr. Cheryl Rosel B. Ariola, to Dr. Ricardo P. Babaran, to Dr. Michael C. Villarde, and to Dr. Jinky Lailani D. Lu.
To formally close this year's research symposium, please help me welcome the Assistant Vice President for Academic Affairs Research and a professor from the National Institute of Geological Sciences, UP Diliman, Dr. Carla B. Malanta.
Hello, good afternoon po everyone, and we are very grateful to everybody who attended the 2-day symposium that we organized. We have had very insightful presentations and we have very interesting discussions among our participants and our speakers. So we hope that the participants who attended the symposium, you have become aware of the types of research projects that are being conducted by our faculty and researchers from all our different constituent universities. So as we bring the research symposium to a close, we wish to thank our Cycle 8 project leaders. Despite all the challenges of the pandemic and the lockdowns, they were able to produce the results to their respective project teams. The participants who joined us for these past 2 days, our UP officials, and of course, the valuable insights from our EMC. So once again, we thank you for being part of the university's research undertaking, and to our project leaders and project teams, we wish you all the best in your research endeavors and may your tribe grow. Thank you very much.
Thank you, AVP Carla. Thank you, everyone, for joining us this afternoon. We appreciate your support and attendance in this year's Research Symposium. I would also like to invite everyone to please answer our feedback form. You can scan the QR code presented on your screen or access the link posted on the chat box. For those who would like to receive their certificate of attendance, this feedback form is required. Again, I am inviting everyone to please answer our feedback form by scanning the QR code or clicking the link posted on the chat box. If you would like to receive your certificate of attendance, this feedback form is required. Before we all leave the room, may I invite those joining us in the Zoom to turn on their camera for a group photo, please. We have 6 slides. All right. Slide 1, smile, please. 1, 2, 3. Screenshot. Slide 2, 1, 2, 3, smile. Slide 3, 1, 2, 3, smile. 4, smile. 5, and 6. We are ending the 2022 Research Symposium with the singing of the University Hymn, UP Naming Mahal. And that ends the 2022 Research Symposium. Thank you so much for joining us, and we hope to see you in our future events to feature the university's research projects. Keep safe, everyone, and goodbye.
Thank you, everyone.
Bye.
Thank you. Thank you.
Congratulations.
And we would like to request our
Congrats.
MCs and the organizing
Thank you to our OVPA staff.
Salamat po. Happy weekend po
Congrats.
sa lahat.
Thanks to our project leaders.
We are kindly
Teams.
requesting our EMC po and the organizing committee to stay for a while for a group photo, please. So we let everyone leave the room and then stay for a group photo.
But thank you very much to the EMC for all the hard work.
Ngayon ko lang ata nakita si Fatima.
Ngayon lang po ako nagpakita.
Oo.